Welcome everyone to the workshop on gene editing that is organized by the Committee for Advanced Therapies with the support of the Alliance for Regenerative Medicines. I will now open the floor for some introductory statements from our Chief Medical Officer, from the Chair of the Committee for Advanced Therapies, and from Tim Hunt from the Alliance for Regenerative Medicines.
On behalf of the European Medicines Agency and also of myself, I want to wish all of you, speakers and participants, a very warm welcome to this workshop on gene editing organized by our specialist committee, the Committee for Advanced Therapies, with the support from the Alliance of Regenerative Medicines. Advanced therapies, or ATMPs as they are called in Europe, have always been at the forefront of innovation, providing new treatment options to patients, and some of these products even come with a promise to be curative. EMA scientific committees, and certainly the Committee for Advanced Therapies, have shown to be able to cope with innovation. They are seeing novel products throughout their development onwards into the final stages towards a marketing authorization. Gene editing products is no exception. We have approved the first product, Casgevy, that contains cells that are ex vivo modified using gene editing tools. And the field of gene editing is moving fast. Many new ex vivo and in vivo gene editing products are currently under clinical development. We have organized this important workshop to take stock of the latest developments in the field of gene editing. It will give the opportunity for direct interaction between regulators and the developers, independent on whether they come from academia or industry. And this interaction will allow the committee to understand where the need for scientific guidance is and where regulatory support needs are greatest. I hope that today's discussion will be fruitful and that it will contribute to our common goal, namely to ensure safe and efficacious novel products are reaching the patients. That is the goal of everybody here today, and that's the spirit of this workshop. I will finish this short welcome here, but not without thanking colleagues Dolce Rogers, Patrick Seelis, and Esther Shinclana for making this workshop a reality. I wish you all the best for today, and I will hereby like to pass the floor to the eminent row of speakers in the field of gene editing coming from both Europe and the United States. Many thanks.
Good morning. In my role as current CAT chair, I want to also welcome everybody who is participating online and in person in the room to this event. I think what needs to be emphasized is that regulatory oversight is a collaborative effort, and therefore I also very much thank the Alliance for Regenerative Medicines for contributing to this event. The goal is to come to meaningful oversight by sharing the knowledge on gene editing, which currently, first of all, is not that extensive, and second, is geographically not evenly distributed. And therefore, it's even more important to come together, share our experience, and use that experience to generate meaningful guidance as a next step. So thank you very much for everybody who's actively contributed to this workshop and will contribute today. And I hand over to Tim Hunt. Thank you.
Good morning. My name is Tim Hunt, and I am the CEO at the Alliance for Regenerative Medicine, or ARM. As some of you know, ARM is a 400-plus member nonprofit organization made up of biotechnology companies, leading academic centers, and patient organizations. This morning, I really want to thank Emer Cook, the EMA team, and the CAT for co-hosting this important forum with ARM, and for the very strong collaboration that we have developed. At ARM, we greatly appreciate our partnership with Emer and the whole EMA team. Now, we're very excited about this workshop on gene editing, which builds on a November 2024 scientific exchange that we co-hosted with the FDA in the United States. Through regular discussions, bilateral exchanges, and partnerships, we are shaping practical solutions together in service of our patients, far too many of whom face death or serious disabilities from rare genetic conditions. At ARM, we really appreciate this approach, finding specific and actionable things to work on, rolling up our sleeves with partners, and getting after them to help our patients. Today's workshop is exactly along these lines, and it reflects not only our shared commitment to move from scientific promise to regulatory practice, but I would argue it reflects our shared values to truly help patients in need. Now, digging just a little bit deeper, why does this workshop really matter? This is the first structured multi-stakeholder dialogue in Europe focused on gene editing platforms, safety, and risk-appropriate CMC. It's a genuine opportunity to explore what fit-for-purpose regulation looks like for both rare and scalable applications. It'll build a robust scientific foundation, as sessions will establish a common baseline on safety, off-target assessments, and editing outcomes. It will explore platform opportunities. Case studies will show how prior knowledge and shared data can reduce duplication and accelerate development. And again, we like it because it's specific and actionable. So our goal is really to leave today, not just with a good discussion, which is important, but with clear takeaways on how to advance risk-appropriate frameworks for gene editing. At ARM, we look forward to continuing this important collaboration with the EMA and the CAT to advance Europe's role in innovative therapies. And with that, let me simply thank the speakers in advance, both from the academic setting and from the biotechnology industry, for sharing their practical insights. And to everyone, I wish you a highly productive workshop. Thank you.
Okay, and these introductory words, I will give now the floor to Emily de Vries. Short introduction on what is the scope and the aim of this workshop. Emily, floor is yours.
Thank you, Patrick. Good morning. Also a warm welcome from my side. My name is Emily de Vries. I'm the Dutch member of the Committee for Advanced Therapies. I'm here together today also with the other members of the organizing committee, Alessandra Renieri, Martin Oleksiewicz, Tineke van den Hoorn, Rune Kjeke, and Marcus Timon. Gene editing has special attention from the committee since, as of this year, gene editing is included in the work plan of the CAT. The field of gene editing is evolving fast, with many therapies reaching clinical trial phase. As we've got only one ex vivo gene editing product approved so far, regulators are, similar to developers, walking in uncharted territories. The aim of today's workshop is to gather information on challenges developers face during product development. This input will help us identify topics for which regulatory guidance is needed. Providing regulatory input on posed questions is out of the scope of today's meeting. I'm looking forward to an interactive and inspiring meeting. Thank you.
Thank you, Emily. And without further ado, I propose that we start with the scientific presentations that we have lined up for this morning. So we're a little bit ahead of time, so the speakers also have a couple of minutes more to give their talk. After the presentations, you have time and raise your hand to ask one or two questions. But we have, just before lunch, we have a Q&A session planned. And then you will have the possibility to ask more in-depth questions on the whole field of gene editing. I hope that Professor Urnov is online and that he can present his slides. I think, Esther, he wanted to present himself, if I'm correct. Professor Urnov, floor is yours.
Morning, folks. Many thanks for the invitation. I'm honored to speak with you today about editing. We have a unique opportunity and a sense of urgency for gene editing. That's what I'll speak with you about today. Here are my disclosures. All of them are relevant to what I'm going to speak with you about today. As Gedi was just mentioned, I want to remind us all how well CRISPR gene editing works clinically. These are data from Vertex. These are about 40 subjects worth of swim plots. Each line is a subject. Each unpleasant-looking black diamond is a severe episode of pain. Subjects are lined up based on when they received their own gene-edited HSPCs, and once they do that, it's really remarkable how robust the medicine is in terms of eliminating the vaso-occlusive crises. This was the basis of approval data. And of course, people have received this, and that's Victoria Gray, our hero, the person who was the first one on the trial. As a professor at UC Berkeley, it gives me great blue and gold pride to point out that the scientific foundations for this were laid by Jennifer Doudna together with Emmanuel Charpentier then at the University of Vienna. They invented gene editing after having figured out how Cas9 is targeted to its DNA sequence by a 20 nucleotide stretch of an RNA it carries. It's just remarkable to me that in 8 years, the field went from this drawing in a science paper to a medicine, and look how congruent the drawings are. Really exciting. What's truly astonishing to me is how quickly the field reduced to practice this concept of programmability. That is, swapping out the 20 nucleotide sequence gives you a different medicine. These are data from Intellia, early data on transthyretin amyloidosis. You can see the overall architecture of the medicine is exactly the same, except it's no longer blood, it's liver. The y-axis is serum levels of a toxic protein that ultimately leads to death of subjects such as Patrick Doherty, whom you see here. And after a single administration of a lipid nanoparticle with mRNA encoding Cas9 and a guide RNA targeting the ATTR gene, the serum levels of this toxic protein plummet. And this is now in a phase 3 trial enrolling 700 plus subjects. So honestly, look, here we are, 13 years into the age of CRISPR, multiple trials ongoing, one medicine approved, 2 phase 3 trials. Looking at the efficacy data for sickle, for thal, for transthyretin amyloidosis, and hereditary angioedema, I think all of us in the field would agree that gene editing has frankly exceeded expectations. As a one-time and a curative is a difficult word to use because there may be some symptoms left, such as pain from a vascular necrosis in sickle subjects. But it's worked remarkably well. I mean, we should all congratulate each other very sincerely. But I don't think we should be congratulating each other for very long. Because the following paradox exists. On the one hand, since Jennifer's and Emmanuel's discovery and invention, the genome editor's toolbox has undergone a super bloom. And here's what I mean by that. At the top right is classical gene editing, starting with Maria Jason in 1994, where a double-strand break creates small insertions and deletions. This is the basis of Casgevy. This is the basis of the 2 Intellia trials. And of course, work from David Liu has given us additional ways to make genetic changes, make point mutation repair using base editing, making more versatile changes using prime editing. And this is just a fraction of the overall toolbox. So really exciting scientific developments, and all of this is in the clinic, and we'll have, I think, folks from both Beam and Prime speaking later with us. But here's the fairly difficult truth we have to face. And again, I'm sorry, I know this is only 10 in the morning, but here's the difficult truth. And the difficult truth is that the publicly held for-profit biotechnology companies in this space have really small pipelines. I put together this table for a review I wrote in October last year, and you can see that, out of the 5,000 genetic diseases, this is what the publicly held sector was working on. Not a lot. This table should be larger. Unfortunately, this table has shrunk in the sense that Editas is no longer working on sickle and Prime is no longer working on CGD. And instead, this is as of yesterday, Editas is also working on hyperlipidemia along with Verve. And Prime, as I mentioned, has abandoned CGD, but now is working on alpha-1 antitrypsin deficiency, just like Beam and Intellia and Wilson's disease. So what's going on? Well, I'm going to explain to you what's going on using an example of this arginine 83 to cysteine mutation that's the focus of Beam's effort. And so here we go. Let's get quite technical quite soon, but I hope this is going to be good technical, not bad technical. Before I explain to you what's going on, I've had the good fortune of having started on gene editing in 2002, so I can I have a 23-year track record of monitoring progress of technology in my field. And I can tell you that this pipeline contraction is not driven by CRISPR technology limitations. As we know from data from a large number of companies, the ones I mentioned and others, the challenge of actually clinic-grade gene editing a target with robust potency and robust specificity has been equally robustly addressed. You know, Prime looked at 5,000 off-target sites for their XCGD program, and they didn't find anything. That's quite remarkable. Okay, so the frustrating thing is people say, well, it's all about delivery. But folks, the bleak truth is, even in fields where delivery is completely solved, there are no trials for gene editing. And let me prove that to you. The little table on the left is, I think, about 10% of a 7-page table of inborn errors of immunity. Each line is a disease, and the number is how many subjects are known to the Model Foundation to be living with it. So we have more than 100,000 people, a lot more, living with these diseases. Their list is growing over time. So every year, scientists do sequencing on people presenting with inborn errors of immunity, and the number of these subjects grows. And finally, as I mentioned, delivery is solved. You can use the Casgevy manufacturing process to make gene-edited autologous hematopoietic stem and progenitor cells for most of these folks. There are no gene editing trials for a single one of these conditions, with Prime having stopped its XCGD trial. None of these 505 diseases is in the clinic for gene editing. Let me walk you through why. It's not often, folks, that I can look at a problem in the real world and say, aha, there is the culprit. Well, folks, here is the culprit. Let me be clear. I will mention Beam's program. I am speaking 100.0% for myself, and I'm not representing Beam in any way, shape, or form. If I misrepresent something, my apologies. Please correct me later. The table you saw earlier, Beam is working on arginine 83 cysteine that causes glycogen storage disease 1A. Let us look at that mutation, and this looking will be informative. Here it is. This is a genome browser. Here is the sequence of the gene and the amino acid it encodes. The little unpleasant looking white, red rectangle, sorry, it's early here, I apologize, is the mutation. And that's the mutation they're working on. And the very first thing you'll notice is there are other mutations in this same stretch of the gene that they could be repairing to treat this disease, but they're not. It gets more interesting when we zoom out. And this is the entire gene, and every little red line is a mutation. So there are a lot of other mutations in this gene that cause this disease. Beam isn't working on them either. Now, if we zoom out to the entire human genome and the known clinical landscape of glycogen storage disease, you will see that there are 8 different genes, in genetic speak, 8 complementation groups, mutations in which cause GSD. But again, they're not in the pipeline of Beam. It's a prevalent mutation they're working on, but it's not the single one. What's going on? Okay, so here is what's going on. I'm so grateful to the EMA for gathering to speak about regulatory, because here is the problem. This is the centerpiece of the issue. The medicine you approved and that the FDA and the MHRA approved, the vial of Casgevy has a single chemical entity, well, actually 2, that can be used to treat any person living with sickle or thal. It has Cas9, a protein, and 100 nucleotide specific guide RNA with a specific 20 nucleotide spacer that targets the GATA1 footprint in the plus 58 DNase 1 hypersensitive site of the BCL11A erythroid enhancer. Every single person gets gene edited cells modified with that chemical composition. But by definition, for Beam or Prime or any other company working to repair mutations in glycogen storage disease, every single guide will have to be different. And in some cases, you might even have to use a different enzyme. It's the same disease, but every editor needs to be specialized for the person. So this means that Beam right now, under the current system, is working for one mutation, one editor. But the problem is that they're going to spend tens of millions of dollars, which is how much it costs, a lot of CMC, to get to approval. But after all of that work and all of that spend, they will only be treating a fraction of the patients living with this condition. This is why the for-profit sector is largely working on alpha-1 antitrypsin deficiency, sickle cell disease, and cancer, and cardiovascular disease, because these are prevalent. So you can use the same guide and the same editor for everyone. But unfortunately, as I mentioned to you, that leaves a giant swath of Mendelian disease, tractable Mendelian disease, off the table. For example, inborn errors of immunity. This is what we're going to fix. We need to split this problem into subunits. And I've represented them as stacked dolls, the outermost problem and then the innermost. And the innermost is the most toxic one, which we're going to discuss. So under current rules, one mutation is one IND or one CTA. This means that one editor is one BLA, one approval. This means that right now you cannot have multiple editors in the same IND or CTA. And unfortunately, when you go from one guide to another, you have to redo studies that actually do not need to be redone, and yet they are redone. So enough hand-wringing. This is now, and let's transition out of sort of lamentation mode and into problem-solving mode. We have to be able to pool patients by syndrome, and we're actually doing that, and I'll explain how. We have to put people with the same clinical presentation of glycogen storage disease under the same IND or CTA. We have to be able to put multiple editors in one IND. Look, I know that's not what's currently done, but what I'm communicating to you is we have to do it. And if we don't do it, we will be speaking about this problem for the next 20 years. And finally, we have to leverage platform processes in PharmTox and CMC. And what that basically means is you do not redo redundant studies. In speaking about this platform challenge, something that has been a passion of me personally and the Innovative Genomics Institute, where I worked for the past 8 years, I've struggled a bit of how to represent what is a platform. People argue, what is a platform? Well, let me simplify this. A platform is this. Let's say you're treating a genetic disease and you've built an editor for one person, and it costs you a certain amount of time and money to go from 0 to 1. And that's the unit of effort and money. Platform is when you go to treat a second subject with that disease, even if you change the editor a little bit, going from the first subject to the second is less time and money than going for the first one. So 2 minus 1 will be less than 1. A platform further means that when you go to treat a third subject where you keep everything the same, but let's just change the 20 nucleotide in the spacer of the guide RNA, that costs even less money than going from 0 to 1 subject and less money than going from 1 to 2. This may seem primitive. I'll show you in just a bit how we're actually trying to reduce this to regulatory and clinical practice. And I know many people later today will speak to the same. Let me point out, and again, I'm not speaking for Intellia, but I'm representing their data as best as I can. You know, Jennifer and Emmanuel invented this editing approach where you swap the guide and you get a different cutter. Well, and Intellia did that, but for the clinic. They delivered a guide RNA and an enzyme to disrupt ATTR for transthyretin amyloidosis, and these are early stage clinical data. They're in phase 3. Then they swapped out the 20 nucleotide spacer. The mRNA encoding the enzyme is the same. The excipients of the lipid nanoparticles are the same. The formulation is the same, and the route of administration is the same. It's a completely different gene on a different chromosome, and they're also in phase 3. So this has been done. Now we just have to do it over and over again as much as we can. How to do that? Well, remarkably, the not-for-profit and academic sector has had to step in. I work for Jennifer Doudna. She enunciated a vision for all of us working for the IGI that CRISPR has to become the standard of medical care for certain diseases. I respect the right and the imperative that the for-profit sector has to satisfy fiduciary requirements to their investors, which is exactly how the world works. We are UC Berkeley. Our mission is Fiat Lux, let there be light. And so our goal is to do research that is, if you wish, a rising tide that lifts all boats. Let me speak to the practicalities here. There's a lot of people I need to thank in presenting this. First and foremost, Danaher, my colleagues at Danaher, including Sadiq Qasim, who will speak later. My colleagues at CHOP Medicine, Kiran Musunuru and Rebecca Barnes-Nicholas, who will speak later. The wonderful, wonderful people at the ARM, including Mike Lemicke. There was a scientific exchange with the FDA, and the outcome of that has been published. A spectacular lineup of folks from non-for-profit and for-profit companies contributed to both. Thank you, thank you, thank you. I will simply refer you to both of these papers and the fact that a number of these folks will speak. So much as I'd like to pontificate about this, I won't. I do just want to point one simple thing, that we've proposed something very simple and actionable that we're actually reducing to clinical practice. So if you read this piece, you will see this table where multiple groups, whether they work on, as we do, hemophagocytic lymphohistiocytosis, Kieran Musunuru and Rebecca Ahrens-Nicholas will later speak to their work on urea cycle disorders, and so on and so on. So all of these folks contributed. But the foundational concept of platformizing CRISPR and expanding its footprint means this. I mentioned today, Casgevy, one guide, one enzyme for everyone. As I mentioned, this does not cover and prevents, in fact, the clinical development of medicines for the vast majority of Mendelian disease. We argued to the agency that you should be able to have multiple guide RNAs using the same enzyme, as long as it's the same target gene. So, for example, you know that GSD1 mutation landscape? If you're using the same editor and swapping the guides, you should be able to put them under one IND or CTA. Next, having de-risked that, you should be able to put different editing modalities. So double-strand breaks, base editing, prime editing, something else editing, as long as you're still targeting that gene and that clinical picture. And finally, we argued you should be able to expand to many genes. So, you know, the 8 genes that cause clinical symptoms of glycogen storage disease. The hope, of course, is that the more you do that and the more you bucketize mutations under the same IND CTA, the more patients you'll be able to cover. I want to emphasize that none of this is scientifically, how do I put this, difficult. I mean, and people will say, well, how dare you? Editors are difficult to design. They are, but they aren't. You just assemble them and test them and you find the one that works. I mean, 12 years ago, this was really hard. 6 years ago, this was hard. Today, it's a lot of work, but it's not hard. It's not. There is one other component in addition to this bucketizing of mutations under the same IND or CTA. And that other component, which we advocate for very strongly and are reducing to clinical practice, is this concept of benefit-risk proportionality. This is something that Tim Hunt mentioned. So what does that mean? Again, I've been speaking about this for quite some time now, and this is the best I can offer. And I'm really indebted to the amazing physician scientists and physicians who are on this slide for teaching me about this. So Rebecca Ahrens-Nicholas, who I think will speak later, the rest of the physicians on the call are close colleagues working with us on inborn errors of immunity. They're at UCSF, UCLA, in the United Kingdom. What's a benefit-risk appropriate path? So if a physician such as this one or any other has an open IND CTA for CRISPR gene editing in severe pediatric disease, this could be a devastating error of metabolism, as in the case of CHOP, or a devastating error of the immune system, as in the case of UCSF, UCLA, or Greater Ormond Street Hospital. If you have a newborn diagnosed with a new mutation, you should be able to develop an editor before the kid dies under the same IND. It's that simple. Let me give you a clinical story. This is from Michelle Hermiston. This is a child who died of a mutation in PRF1, which encodes purpurine, and the disease is called familial hemophagocytic lymphohistiocytosis. Dr. Hermiston told us that this was a 26-month-old boy. He was well, got a runny nose. Oh, well, alert the media, runny nose. But unfortunately, in the hospital, he started seizing and developed devastating symptoms. He was diagnosed with this terrible genetic disease, and he died at the best tertiary care hospital for this disease on the west coast of the United States. So I'm not a medical doctor. I can't speak to this, but I am an editor. And I can tell you with definitive certainty that it is technically feasible to engineer and de-risk a gene editor to repair this mutation within the timeline of this child's life, to gene edit the patient's T cells and give them back to the patient, even though we had 42 days. The concept of benefit risk is powerfully illustrated on this slide. These are data, and I'm so sorry I forgot to drop the reference. I will fix the slides later. The standard of care for HLH is etoposide, which is a horrific chemotherapeutic. It creates an enormous amount of double-strand break by inhibiting topoisomerase 2. The pharmacokinetic profile of etoposide is that children treated with etoposide have about 20 micromolar etoposide in their circulation for a month. These are double-strand breaks enumerated in cells growing in culture after being treated with that amount of etoposide. Every shiny little dot is a double-strand break. Look at this image and tell me, in what way is it meaningful to look for off-target sites of a gene-editing nuclease when the child has been on a month of a drug that creates that much double-strand break burden throughout the child's circulation? Well, I'm an editor, and I'm going to argue that we need to fundamentally new way to de-risk gene editors if we are trying to save a kid like that. That's benefit-risk proportionate regulation. The reason this is necessary is just like GSD or many other genetic diseases, this disease is heterogeneous. It's caused by mutations in 6 different genes at least, and every child presents with their own mutation. So if the goal is to save this kid, we have to work in a fundamentally new non-clinical de-risking framework. So together with a magnificent team of UCSF and IGI scientists and physician scientists, and with magnificent support that I'm very grateful for from Danaher, we are directly reducing everything I said in this Stackdale image to regulatory and clinical practice at UCSF, with the understanding that 30% of newborns with familial HLH have 2 to 3 months to live. We are building a clinical trial where we can enroll subjects with mutations in multiple genes. In close partnership with Danaher, we are establishing a CRISPR on-demand gene editor design and de-risking pathway where we can get to a clinic-grade editor for gene editing in T cells within the timeline before the kid dies. One key component of this de-risking mindset, specifically with respect to off-target sites, we just have to remember this kid is on etoposide. Finally, this make new editor on demand for the next child leverages heavily the platform concept where, for example, we reuse the same manufacturing scheme. I know the organizers graciously gave me an extra few minutes. Let me retain that time advantage by wrapping up. I'm honored beyond words. The most important experience of my professional life was being led by the amazing team at Chop and Pen and working with Sadik, all of whom will speak later, to contribute to the development of a medicine for Kyle Jr. I'm not going to speak to the science or the clinical story at all because others will better than I can, but I will speak to my lived experience in the United States of the difference this child has made since Rebecca, Kieran, Sadik, and yours truly, and a magnificent group of people described the story in the New England Journal in mid-May. In early June, the leadership of the American healthcare sector, and I know some of you may not know what these acronyms mean. These are basically the folks who regulate healthcare policy in America, including the head of the FDA and most importantly, the head of the CBER, the Center for Biologics Evaluation Research that regulates us. At that workshop, David Liu and yours truly spoke extensively about baby Kyle Jr. and the essentiality of advancing this to other American children. Secretary Kennedy said that CRISPR gives the gift of life, and I agree with the secretary. He's correct. It does. But what was striking is the next day, there were all these social media postings about, we really have to do more of this. And all of us, frankly, who work on this were amazed in a good way. And then Health and Human Services posted this message. And, this could have stopped at this message, but that is not what happened. My lived experience has been that the leadership of the FDA and the leadership of the CBER has proven remarkably eager to listen to us in terms of what exactly this specific sector, CRISPR on demand for severely ill human beings, will need from a regulatory innovation perspective in order to advance more such approaches to the clinic. Rather than dive into the details, I'm just going to read to you a page of text. I know this is a lot of text, but I promise you, please stay with me. The last paragraph of what I'm about to show you will blow your mind in a very good way. The director of the CBER and the commissioner of the FDA are about to publish a piece in the New England Journal of Medicine. And in that, they describe a new FDA approval pathway for N-of-1 therapies. The vision here, which Director Prasad enunciated in a presentation that was reported in Endpoints, is that there will be a new approval pathway for those with extremely limited treatment options. The director and the commissioner proposed to call it the plausible mechanism pathway. What that means is if you have a genetic disease and you're directly repairing the mutation in a way where you can directly connect improvement of patient symptoms with repairing the function of the gene, whether you're fixing your urea cycle disorder or hemophagocytic lymphohistiocytosis, if you fix the enzyme that metabolizes nitrogen, or if you fix the protein that causes T cells to function normally, then we have a plausible mechanism pathway for how the drug works. It will be a novel pathway. By that, I mean individualized therapies that baby A gets one treatment, B gets the next, C gets the next. The commissioner closed, the director closed his public presentation about this in saying, quote, "The upcoming New England Journal publication will provide drug makers with a roadmap for how they can win approval for such therapies after treating just several kids or young adults or adults with a certain condition." He added that these approval trials could be as small as 5 people. Look, wrapping up, 60 seconds. We have to get to the clinic more in order to get to that world. The only way to find out how to safely edit people is to edit more people. The 36-year history of cell and gene therapy has near no examples where severe adverse events due to the treatment were identified in non-clinical studies. These are just 4 examples recently where that's true. The iatrogenic leukemia on the Bluebird trial for sickle, the death of the child on Tim Yu's personalized antisense, 2 recent deaths on AAV gene therapy trials, one with an established serotype, one with an innovative one. None of these could have been predicted from any non-clinical experiment. This means that it is imperative we gain more information from the lived experience of subjects on clinical trials for personalized CRISPR. That information will tell us where the true risk, if any, lies. Finally, I always choke up a bit when looking at Rebecca's patient. I think that's Rebecca holding him, and that's, I think, Kieran's hand extending that. Rebecca and Kieran will speak to this. This was May 2025, folks. Look, in 3 years, what will we be talking about? Well, I really hope that we'll be talking about multiple children across jurisdictions, whether in the United Kingdom or the European Union or the United States, regulated by different regulatory authorities where they will have been treated via this accelerated pathway. I think it's completely realistic to get to a sizable cohort of such children across jurisdictions. Finally, I know that there's a lot of effort in harmonizing regulatory approaches. I think this specific area, platform approach to CRISPR on demand for devastating disease, is uniquely amenable to harmonization because baby Rosalind of London or baby Gregor of Brno or Barbara from Cold Spring Harbor, they do not wear their country's flag on their little gown that they're wearing in the hospital. They just have a genetic disease. So I really encourage the regulators to do all they can to harmonize and provide frameworks for platform approaches for CRISPR on demand, such as in 5 years, and going back to the vision enunciated by the director of the CBER, there will be platform approvals for hematologic, metabolic, pulmonary, and dermatology indications, such as epidermolysis bullosa, for example. The critical point being, each one of them reduces to regulatory and commercialization practice this concept that you can and should be able to make an approved medicine on demand. Thank you very much for the honor of having me open this remarkable workshop with a scientific presentation. I will close on this. We are working actively to reduce this to clinical practice. We have support that we're thankful for from Danaher and from the Chan Zuckerberg Initiative. We are doing exactly that and reducing this to clinical practice and sharing the non-clinical and clinical data broadly. But ultimately, I am so excited for the multiple efforts of this type that are going around the world. And this will take courage. And remember, courage, as Andy Rooney reminds us, is not being fearless. It's being scared to death and doing the right thing anyway. Here, I encourage all of us to have that kind of courage to do the right thing by our patients. Thank you.
Thank you, Professor Runo, for this really inspiring presentation. And just a small comment from my side. In Europe, there is no legal obstacle to having multiple chemical entities in a single clinical trial. So while we don't have experience and while we certainly need to work on it, the situation is a little different compared to the US. So what we maybe should focus on is how to generate the scientific background for leveraging pre-existing data and how to best define platforms going forward. But there is not quite the same urgency in defining platforms to enable clinical trials as there might be in the US. And with that, I open the floor to questions. Alessandra?
Hi. Yes, I have a comment more than a question. So I'm very pleased and I appreciate very much this introductory talk by the Dr. Urnov, the colleague from Berkeley, concerning these two main points that are the mutation heterogeneity and the turnaround time for approval. Concerning the mutation heterogeneity, we already discussed in the CAT in the past, under the leading of the previous leader, Martina Schulzer, the idea to have an approval of a basket of mutations. So I'm sure that now, under the leadership of Ilona Reichel, we can discuss again this in more practical way. And so thank you so much for pointing out this, that I agree. These are the two key points for moving forward.
Thank you.
Are there any further immediate questions? If not, we can leave further questions to the discussion later on and move on to the next presentation. Patrick?
So many thanks, Ilona, and also my thanks to Dr. Urnov. Our next presentation is on the next topic that we want to have the input from the experts, and this is on how to evaluate safety concerns. We have two presentations lined up. The first one is by Dr. Naldini, and I give him the floor for this first talk.
Thank you, Patrick. Welcome, and thank you, Ilona, the chair committee, and the people are listening. And again, as mentioned, I will try to discuss some of the effort to deep analyze this, the potential outcome of genome editing technology. Now, we have a plethora of genome editing tools. These are modular machinery, essentially made by several components. So they do have a DNA recognizing domain. We have a number of them. Of course, most of them today, and most of my focus presentation will be on the Cas-derived enzyme, which use a guide RNA to tether the editor to the specific target sequence. And then they have an enzyme component, which varies according to the intended outcome, endonucleases, deaminases, reverse transcriptases, epigenetic modifiers as well. The intermediate of the action of this enzyme at the target site are a double-stranded break. For the nucleases are base changes and nick for the base editors, which essentially are dependent on a nickase and not a full nucleases. And intermediate bearing flaps and nicks for the prime editors. The outcome of that intermediate, which involve the engagement of different type of repair processes in the cell, is a sealing of the break. In the case of the nucleases, this may also include indels, so insertion or deletion of small or also large fragment of DNA. In case of homology-driven repair, will be the insertion of the template, which is co-delivered with the nucleases. Base editors will have a fixing the base changes at the target site, and prime editors will have a new sequence, usually short sequence, inserted at the target site. Now, in terms of assessing the specificity of these editors, most of that really is dependent on the guide, which is the RNA molecule in case of the Cas-based approaches. We have quite an established approach to investigate guide-dependent specificity. Usually these start from an in silico analysis of the uniqueness of the target sequence and limited representation of its homologue throughout the genome, which are then ranking in terms of course, a level of homology. This usually end up with a very large list of candidate off-target site, which are investigated by direct in an experimental manner by a number of different technology. We have, again, a growing list. These are quite well established. Some of these are working on free DNA and others directly in cell, usually cell line, which would better represent, of course, the chromatin. Again, this technology end up with an improved potentially list of candidates, which are then nominated to a certain extent ranked. Eventually, however, the top candidate from this usually long list are validated for effective potential of targeting into the cell of interest and also in the delivery condition of the editor of interest. While this is quite a very established approach, I must say that there are some issues in terms of the cost and the burden of this analysis. They usually have a very long list of potential of target, very few of which are usually validated in the experimental condition of choices. There's also limitation in the sensitivity of those validation approaches. Of course, I think there is no rationale for aiming to an exhaustive validation of all potential targets. This will be unrealistic. And essentially, while we may be satisfied with some level of assessment, probably one of the other advantages of this long list of candidates is that if there is some potential risky of target which appears somehow in this analysis, like for instance a p53 target sequence, potential of target, of course you would discard that guide even if there is no proof of validated targeting. We have been developing additional assays which are more unbiased in terms of genome-wide, rather than focusing on prediction of or experimental, essentially, nomination of candidate of target. These are far from being a routine type of assay. They are mostly relevant to validate some of the process on the guide I will mention later on. I also want to mention that there are additional issues to consider. The human genome variation, of course, which is hardly represented into the database that we are using, which requires some attention. And of course, the purity also, the guide RNA. Emerging evidence are shown that in many cases, this molecule are not as pure as expected, certainly for experimental use as well. Functional significance is always a very important point because, of course, again, most of those potential of target may not be biologically also relevant in terms also of safety prediction, may be found like in intron or sequence, which are not known. There is another area of complexity which are emerging more recently, is the realization that the outcome of the DNA repair at the target site is far from heterogeneous than it was originally expected. Of course, in case of non-homologous enjoining another type of repair, sealing of the break, there are a number of potential sequence variation, including very large sequence that somehow escape some of the early studies in terms of the substantial frequency of this event. There may be trapping of material, including exogenous DNA and viral vector, for instance, derived DNA for the template. And we're aiming to some more complex repair pathway, like homology-driven repair. Again, we may have the intended homology-driven copy and paste of the template at the target site, but there are also a number of heterogeneous outcomes, including partially homology-driven repair and non-homologous enjoining on the other side of the break, aborted homology-driven repair with just a partial fragment of the template, or full trapping of the template and molecules at the target site. Again, here, how do we deal with the analysis of this heterogeneity? We have a number of assays which are well established in terms of assessing the extent of editing at the target site. This is usually small edits which are captured, and these are routinely used. A little more effort requires to investigate the occurrence of large deletion. Of course, large deletion means you are missing essentially your target sequence, so you may entirely miss the occurrence of that large deletion unless you use a more broader assay like tiling the loci flanking the target site by PCR, targeted PCR, or some sort of long sequencing or optical or electronic mapping of that locus with a broader view. Again, here we have a cost and time issue. All of these assays do have a question bias. They will only look what you are looking at. And again, in most of the cases, the functional significance of different repair outcome in terms of safety is limited. And again, here I would say if there are potential genes flanking your target site, which could pose a risk in terms of deletion, then that would be something you would flag. Otherwise, most of those repair outcome may be well tolerated, or on the other hand, self-purge away. I will now use our experience of developing a strategy for homology-driven repair of a primary immunodeficiency involved gene like CD40 ligand, causing its deficiency, causing hyper-IgM syndrome, as a sort of a case study of the type of analysis we have been performing to validate our process. So this primary immunodeficiency is due to the lack of function of CD40 ligand in CD4 T cell, and CD40 ligand here is essential to trigger affinity maturation, and so immunoglobulin class switching in B cell, as well as full activation of many innate immune cells like macrophages. This gene is tightly regulated and drawn endogenously, and there is indication that the constitutive expression, like by gene replacement, would pose a risk in terms of the safety of that product. And so a precise repair in situ, which would reconstitute function as well as a transcriptional control, would be far preferable. Our strategy here to deal also with the several mutations, Fyodor mentioned before the challenge of targeting, of course, multiple mutations affecting the same gene, is using a knocking approach based on homology-driven repair, in which a corrective functional cDNA is inserted downstream the endogenous promoter of the affected locus. In that case, the endogenous promoter will drive essentially expression of a functional construct, essentially correcting all mutation which occur downstream the insertion site. Some of the advantage of this strategy in our case is that we did essentially target the first intron of the gene, and so we could essentially use a truncated cDNA, which means that any off-target insertion would not express the full product, and of course there's no promoter in the template, and even imprecise insertion here would probably be functional because the splicing into the exon could tolerate a different type of repair outcome. On top of that, because this is an X-linked gene of a disease, and we couple the cDNA to a clinically compliant reporter or selector like a low affinity NGFR receptor, which would allow to select the edited cell and at the same time purge all the repair outcome. We would not end up with the insertion, the desired insertion and functional reconstitution. So the process has been fully developed at GMP scale and grade for clinical use over a couple of years of work, essentially started from selection of CD40 cell from patient, then electroporation of the gene editing nucleases. I will get it in a moment, together by the template repair, then selection of the edited cell by the NGFR beads, essentially, and expansion and formulation for clinical use. The efficiency of the homology-driven repair is shown here in different experiments. Upon the selection, of course, this can go to nearly purity in the outcome of the selection, and importantly, the increasing growth of the cell upon the final cDNA expansion is robust, leading to very high numbers expected for the clinical trial, and equally for patient and healthy donor cell in the development, and not that different from untreated, unedited cell. There is some initial gap upon editing, which slow, of course, the initial proliferation. The phenotype of the final product is also maintained with a good fraction of a cell with early memory, central memory phenotype. The patient in this disease, they tend to have a higher fraction of those memory T cells because of the block of maturation. So that is an expected feature. You can see here the full reconstitution of the regulated expression of the CD40 ligand, which essentially is associated with activation of the T cell, and then essentially it stays for a few hours and then again is lost. Whether we edit on healthy donor cell, we recapitulate the physiological expression of non-edited cell, and in patient cell, essentially, we reconstitute expression of the gene similarly to normal cell, as compared, of course, to the unedited patient cell to begin with. Our effort to characterize the specificity of our guide was based on 3 orthogonal strategy, in silico prediction, as I mentioned, and then 2 assays, one on digenome of DNA, essentially, and guide-seq performed on cell treated with the nucleases. There was about 115 candidates of target being nominated, which were tested for validation into our editing condition. Only one of these, which was the one commonly identified by all 3 strategies, was in fact identified as acted at a detectable level above 0.1% in the T cell in our experimental condition, moving to a high fidelity Cas9 suppress detection of target, providing for essentially a very specific editing approach. This was also confirmed, as I said, by Cas-seq now in full validation, in which the only site which show essentially activity and insertion or break in this case was the intended site on the X chromosome, and also by a genome-wide mapping. These are 3 products from our validation run full scale, in which the only recurrent, again, insertion was at the target site on the X chromosome. The specificity of this validation assay, in the case of Cas-seq, and also deep sequencing of the identified target, may be in up to 0.01% of specificity. Now, as I mentioned, there is a need to explore a larger deletion at the target site. They would be missed by those assays which only look essentially at the sequencing of the intended target. So we tiled the locus by devising a targeting PCR system on the flanking loci site, about 10 kb on each side. We did find deletion also to significant extent. This was most visible in the case of female cell, in which those deletions were better tolerated than male cell, and they were also aggravated by the presence of a template vector, in this case AAV, reaching up to 30% of deletion in the early day after treatment. The increasing MY of the vector was also increasing that deletion. Switching to IDLV integrase effective lentiviral vector for a number of reasons actually improve that effect. So there was fewer those deletion, and importantly, the selection by the NGFR of the functional edited cell essentially purge away all those deletions. So there's two aspects here. As often is the case, large deletion may be self-purged, as you can see here during culture. And also, of course, the advantage of a X-link editing in this case, coupled to a selection, is the enforced purging of those deletions. Long-read sequencing to validate the editing outcome at the target site did show indeed that the vast majority of the edited cell, again, all of these are NGFR positive now, after the selection, the vast majority bear the intended homology-driven repair edit. A fraction of these are still native, or they have small indels, and the green one actually bear imprecise editing, so maybe like non-homologous enjoining, which, however, as mentioned before, are still functional, so they still lead to expression of the rescued gene as well as of the selector. Optical mapping, so unbiased analysis of those three products that I mentioned, again, did only show as a recurring event insertion at the X chromosome target site. These insertions were larger than the expected homology-driven repair. Indeed, they do represent essentially insertion of the full template or a concatemer of that. So essentially are this green essentially editing event here, in which non-homologous enjoining on trapping may contribute to that part of insertion. But again, as I said, these are functional events. Now, I think I really love this analysis. Again, this was done to validate the process, not to be considered as a routine assay. We did finally got to do single-cell analysis of the genome on the edited, in this case, the selected edited cell. What you're looking here are essentially amplification of loci tiling the X chromosome. You can see here, this is the target site. You see there is in our final product, there is a fraction of cell which maintain that sequence at the target site. All the vast majority have a deletion here, but it's very nice to see the deletion is essentially precise. So there is no larger deletion. As mentioned, we have purged them away. And instead, we have insertion now of the template cassette. Most of the cell have only the cassette, which means they have underwent homology-driven repair. A fraction of those include also flanking sequence from the vector. These are, as I said, those trapping events, which, however, are functional. So I think this is very reassuring in terms of a product which essentially has, for the vast majority, the intended functional event. 95% of the cell are expected to be functional and bearing no large deletion, with some heterogeneity, which, however, has no functional significance. And this is essentially the same analysis on the starting cell. Of course, there is no insertion of any type and there is no deletion. This is the bulk analysis, the product, and then this is the selected one, finally. Now, we also perform Cas-seq, as I said, as a genome-wide assessment for translocation, insertion, and other site. So this assay, developed by Toni Katomelan, essentially amplified by a bait around the target site. But essentially, the target site is intact or bears small indels. There are decoy primer, which essentially prevent the full amplification. So this is essentially takeout, which is the most abundant outcome from the final analysis. Instead, focusing the analysis on those rarer events in which there is no more sequence for the decoy primer to bind, and because there are being like insertion, translocation, of deletion. So this assay allow, as I said, to look for those wanted events. I already mentioned that there are no off-target site event from this analysis. Now, looking at the on-target, we can measure deletion or inversion, in which, again, we lose those primer binding sites, so we have a larger deletion and come up to several kb. This is the product treated only with nucleases. If you have a template like IDLV, as I said, there is a significant lower extent of those large deletion, and upon selection of the product, again, we do have a very highly purified and enriched editing at the target site, or immediately flanking it, while what has been negatively selected will be mostly bearing those larger deletion. So this briefly I mentioned as a case study. I think it's feasible to develop a process which allow in situ gene correction of most mutation occurring at the target site with rescued function and regulated expression. The possibility to couple selection of cell carry the intended edit, of course, provide for a high level of precision in the final product with lower heterogeneity of the editing outcome, and importantly, purging of the any larger deletion or potentially an off-target event. And we expect to really file the IMPD in the next, really actually in the next couple of months, and hopefully start the trial very soon. So what we stand today, well, we do have, I think, an established approach to assess the potential risk of off-target genotoxicity from a guide-dependent editor. This is a little bit burdensome, but very doable. There's way to do that, and I think provide for a significant level of confidence in the approach, because the guide can be very specific and the assay can reach a significant, I would say, level of sensitivity to ensure against substantiality of targeting. There is, of course, much more heterogeneity on target occurring upon a double-strand break. Most of this will be relevant depending on the genomic neighborhood, the type of editing and the target cell. So this is a little bit, I would say, much more, target gene dependent, as most of the event will be biological neutral and even preserve the intended outcome. Unbiased genome integrity assays is, of course, very complex and certainly with low resolution. Of course, we do need to have some level of that in our development, of course, to validate our approach. And most importantly, because of this assay are also often biased by what you design to look for, we have to be careful not to look under the lamppost only where we have light, but essentially looking in an as unbiased manner as possible. Very briefly about base editors. This, as I said, have a smaller scope of changes at the target site, base changes. There's a number of them. They were developed by David Liu, as mentioned. They are extremely powerful and efficient tools. They still depend on a nick, and which means that in a certain fraction of cell, there could be also a double-strand break as a byproduct, not because it is required, but it may occur in the cell. So they still would require some validation of the target site. From that perspective, I would say there are a couple of important points to be considered. Those base editors do bear a constitutive enzyme at variance with Cas nucleases, which is dependent on essentially change in structure once it is bound to the target site, which activate nucleases. The base editor are already constitutively active, so they may essentially change the base at the target site with a probably very low residence time. So indeed, there is evidence that they may have broader of target activity that actually through Cas nucleases. And some attention must be used in the design of the assay to explore that of target site, hopefully not fully dependent on a mock Cas, let's say. And there is a potential concern of a genome-wide increase of those mutations. Improved tools are continuously developed. I think our effort to identify those potential source of genotoxicity really serve the scientists to improve those tools and also guide towards the clinical validation. So finally, we do have a plethora of emerging new tools in constant evolution. This is great for the science. Of course, it's challenging from the regulatory landscape as we have a continuously influx of potentially better version, but also they also need some level of validation. Human genome sequence variation and unbalanced representation of regional differences is a limitation to what can be tested in terms of off-target prediction. At the same time, as I said, there's some level of relevance and what is less relevant in terms of really deep analysis of that of target. Animal models are mostly useless because, of course, they have a different genome, except for guide-independent toxicity-related more genome-wide effect of the editors. Clonality tracking in patients may be challenging. I think in the field of gene transfer, we have made a wonderful use of clonality tracking to really validate the safety of the product in humans. This is more challenging in editing cell. We can do some level of clonality tracking by non-homologous enjoining, but it's essentially because the outcome of editing is mostly shared by different clones. This is limited. And so there will be a lot of need, and I think as also Fyodor mentioned, clinical test is going to be essentially the key way to really investigate in the long term at high specificity the toxicity. Complexity of interaction with DNA, I mentioned, hit and run process makes difficult to trace eventual adverse event, and that's something to be kept in. Multiplexing, which is advocated by many editing products, of course, will combinatorially increase heterogeneity of outcomes, so may require special attention. And finally, and again, I also like to leverage this concept of platform, which I think is a key for all our effort in gene therapy, whether based on different platforms. I think the safe use of these tools, because they are multi-component and modular, may benefit from validation of individual components. Same editor, same target site, changing the guidance. I think so. I will just second the call from Fedor to really help us all together to move along this issue of the platform and alleviate the burden and the cost to make this treatment available to patients. Thank you.
Okay, many thanks, Professor Naldini. It was a very inspiring presentation. So can I just open the floor and see if there's any direct questions or comments? See Alessandra, please go ahead.
Hi, Luigi. Thank you for this very interesting presentation. I have two questions. The first is related to this very interesting and novel homology-driven repair edit. If you think that this could be applied to any genes, to genes also outside of the X chromosome like that you presented, or if do you think there are restrictions for this approach? And the second question is related to the fact that you said, if I understood correctly, that you solved the off-target event using a high fidelity Cas. So if you are suggesting to use this approach for any other approach, if you can share the Cas that you have used in that occasion.
Okay, so for the first question, yes, homology-driven repair has been, I think, used from early days of genome editing as a strategy to really do a gene size edit. So it's currently still the most feasible approach to introduce large pieces of DNA, like a whole, cDNA of a gene. So that is essentially, it requires a break and the cell transiting through SG2 phase for this repair pathway to be available. You can certainly use for autosomal genes. We and others have been using. There's an effort, for instance, to target the CAR-T into the TCR locus. We have a development of a RAG1 gene editing approach in stem cell. So this is absolutely. Of course, when there are two alleles, and this is a general issue, characterization of the on-target outcome at the two allele is more challenging than you have a single one, of course, but this is okay. The high fidelity Cas, I think, again, this is just, I think when you validate your off-target profile, there's different way you can go. Usually you can change your target sequence if unsatisfactory. You can look for PAM-less or PAM variant Cas. There are higher fidelity Cas available on the market. This is the case of the one that we have used, which have been developed. There's a couple of those products. In our case, provided essentially lower that off-target activity. As always, you may pay some price in terms of efficacy, which is minimal, so it's a risk balance. So I'm not recommended to use all over. It depends when it is required.
Okay, Tineke.
So thank you for the presentation. I had a question on the on-target evaluation. So is that also patient-specific, because it may rely on the DNA repair mechanisms? And should you check, for instance, if you have autologous cell therapy, should you check then each batch, or is it sufficient to check some at forehand and then waive it? So should it be included in the quality criteria, or can it be once an evaluation and then the product can doesn't need to be checked for that anymore?
Oh, I guess that's a usual issue. Of course, we do expect some variability, for instance, in the robustness of the repair pathway on the intended outcome. So there's going to be some level of variability. I don't think that there's no reason to suggest that this would have been tested patient by patient. Of course, during the validation of your process, like we did test several healthy donor and patient, and of course you showed that there is a reproducible outcome. So there is variation, but that variation is limited around the intended site. So I don't think there is a reason to do patient-specific pre-assessment of that targeting. And that would be extremely, of course, burdensome. It could be used sometimes, depending again on the way you do, but I don't think it's necessary once you have validated your approach. And of course, those more challenging approach to qualify the outcome are only meant to be used for process essentially validation and not for final product release.
Okay, in sake of time, I propose that any further discussion we will take at the Q&A session. So let me now introduce our next speaker is Dr. Musunuru. I hope you're online, and if I understand, you're going to present the slides yourself.
Yes, that's correct. Hopefully you can hear me.
We can hear you quite okay. Yeah, everything is fine, so ready to go.
Beautiful. Thank you. Good morning, everyone. Thank you for the opportunity to present. I'm actually grateful that Dr. Naldini gave a very complete coverage of off-target editing. And so rather than dive into all the different methodologies one could use to assess for off-target editing, I'm actually going to ask the broader question of what do you do when you actually find evidence of off-target editing? How do you address that? And so the way I have been thinking about it, and my colleagues have been thinking about it, is a framework that I will now present to you. So you've done your identification, your nomination techniques, you have a list of sites, you do some sort of verification analysis in the cell types of interest, and that could involve individual amplicon sequencing or hybrid capture assays or multiplex PCR. But let's say you've actually found something. How worried do you have to be about it? How seriously do you have to take it? Does it actually present biological risk, or is it just true and unrelated? And so I think of 5 points that I consider when evaluating such sites. So the first, probably the most important, is that edit in or near a cancer gene? For example, of the hundreds of genes that have been annotated as potentially presenting cancer risk in the COSMIC database, is your edit near one of those genes? And of course, the reason we worry most about this is because we are concerned about oncogenic risk. If an off-target edit happens near an essential gene that's important for survival of a cell and a cell dies, that's probably not a big deal. We have trillions of cells, and so we have a lot of redundancy. We're concerned about a single cell picking up an oncogenic change that then increases the risk of cancer happening down the road. So that's the first question. The second question is, does that edit affect a genomic site that's likely to have a functional impact? It's going to be more of a concern probably if it's within a coding sequence and leads to a non-synonymous change, but you also have to consider non-coding variation as well. That's just as important. And there are a variety of tools one can use to make predictions as to whether either a non-coding variant or a coding variant is likely to have a functional effect. Things like the ensemble variant effector predictor, the CAD score, which I like very much because it can be used either with coding or non-coding variants and so forth. Third question, is that edit likely to affect gene expression in the target tissue or other tissues in which on-target editing is evident, right? So that becomes very important because it's tied to delivery. If, for example, you're targeting in the liver using systemic administration of a gene editing product, and the gene that lies nearest to your off-target edit is not one that's expressed in the liver at all. In fact, it's not expressed anywhere that's accessible to your drug. Say it's expressed only in the retina of the eye. Well, it's unlikely that that systemic therapy is going to get it to that tissue. So you really probably don't have to worry so much about that. Is the edit likely to occur at pharmacological doses? The actual profile that is presented by the drug in a patient, at that dose, are you actually seeing the editing, or are you only seeing the editing if you're using the supersaturating doses we tend to use in off-target assays? And that's an important consideration because we want to have the highest possible sensitivity in principle. And so we have a tendency to use very, very, very high doses, have very high exposure to the drug product. But as a practical matter, there's no way cells in the body are ever going to be experiencing that level of exposure. It's going to be far, far less. And so we need to take that into consideration. And then finally, this is going to be most relevant to nuclease editing, although as Dr. Naldini pointed out, there is some risk of it as well with nickase editors, whether it's base editors or prime editors or whatnot. Do the various techniques such as genome sequencing or karyotyping or optical genome mapping or so forth, do they demonstrating the structural variation, including the site, any chromosomal changes, translocations, inversions, duplications, deletions, things of that sort. So rather than try to be very prescriptive and say what I think about each of those things and try to, say this is the way you should do things, I thought it would actually be more useful to this audience just to go through a few examples from my experience and the experience of others in this space to illustrate these different principles. So I'll start with this idea of whether the edit is in or near a cancer gene. So this is an actual case. This was a corrective base editing therapy that my academic group was developing, targeting the liver for an inborn error of metabolism. The specifics are not so important. And in doing a very comprehensive off-target analysis, we had one verified site of on-target editing, and it turned out to be an intronic sequence, several kilobases from the nearest exon. Now, the wrinkle is that it was located in the PTN gene, and that, as you're probably aware, is a very, very well-known, well-established tumor suppressor gene, documented as a causal gene in Cosmic, but more to the point, it's actually a very well-characterized tumor suppressor gene, causal gene for hepatocellular carcinoma. So in the tissue type we are targeting. Now, admittedly, it's an intronic sequence, and we're using not a nuclease that would introduce double-strand breaks that might have a higher level of concern. But even with a base editor, there's a theoretical possibility you could get double-strand breaks. There is a theoretical possibility that that intronic sequence might somehow interfere with the protein's function, and any loss of function of a tumor suppressor gene presents risk. And so when I presented this case to a relatively large audience of people, several hundred people, this was at the American Society of Gene Cell Therapy several months ago, and asked the audience, how do you feel about this? If you were developing this program, would you go forward? Pretty much universally across the room, everyone said that they would not go forward with this. And we did not go forward with this because of that, even that sense that maybe there's a little bit of risk for liver cancer. Even if you only getting a deleterious event in a few cells out of the entire liver, it just doesn't feel right. You just don't want to go there. And so we terminated this program. We ended up pivoting to a different guide RNA to make the same corrective edit and have been pursuing that, where this particular gene targeting with the PTEN gene is not an issue. So that was a pretty dramatic example where I think all of us would pretty much agree, no, we don't want to go there. That's just too risky. So the second principle is, does the edit affect a genomic site that's likely to have functional impact, whether coding or non-coding? Is it likely to affect gene expression or gene function, really, in the target tissue or other tissues? And so this is a different corrective base editing therapy, also targeting liver for a different inborn error of metabolism. The details are not so important. And after doing a full comprehensive analysis of off-target editing, we had one verified site. It was, again, in an intronic sequence, several hundred base pairs away from the nearest exon. The gene that was implicated is a gene called ATP7B. It is not a cosmic gene. It does not have an explicit link to cancer. It is expressed in the liver, so you do have to be mindful that there's a potential functional consequence. And it actually turns out that complete loss of function of this gene, it's recessive in a recessive mode, is responsible for Wilson disease, so loss of function of a copper transporter. So potentially a biological consequence if you had high-level editing that actually impaired the function of this gene, but no intrinsic oncogenic risk. And again, we were mindful that this was occurring in an intronic sequence far away from the nearest exon. And when we used the various algorithms such as the CAD score, we found that at least these predictive algorithms suggest that there really isn't anything in the biological risk. So the CAD score for this variant or the potential off-target edit was on the order of 1.2. And if you're not familiar with the CAD score, you really don't start to get concerned about CAD scores until it's 20 or higher. The higher score, the worse it is. And so we went forward with this program. And this program is actually none other than the treatment that was given to baby KJ that Dr. Ernhoff mentioned earlier this morning. So to illustrate the next principle, is the edit likely to occur at pharmacological doses of a drug product administered to patients rather than the supersaturating doses that we have typically been using in off-target assays? One, because we just intrinsically feel that we want to have the highest possible sensitivity. And then two, because there's been something of an insistence from regulatory agencies, and in my case, the FDA, to do those sorts of assays, to look at supersaturating doses. Although I have noticed in recent months they've been backing off from that, perhaps out of recognition that maybe having too much sensitivity is counterproductive. So this example is one from Intellia Therapeutics. I don't represent them in any way. I'm simply talking about this case based on what they have published in the literature. This is Nexigeran, Ciclumeran, or Nexz for short. This is a liver-directed Cas9 nuclease, Streptococcus pyogenes Cas9, for transthyretin amyloidosis. They did a comprehensive off-target analysis, and they verified 7 sites of off-target editing in the target cell type, hepatocytes. And here's data from their publication. This is from 2021, when they first made it known that they actually had what appeared to be a very effective therapy targeting the transthyretin gene, the TTR gene, for an activation, and getting very, very impressive pharmacodynamic results where there was greater, in one case, greater than 90% reduction of transthyretin levels in the blood. And of course, they've now since treated hundreds of patients, and this drug entity Nexz is in phase 3 clinical trials. But from their initial off-target analysis, you see this graph, you see the on-target editing, it shows a nice dose response curve, and then towards the bottom of the graph, you see the 7 sites for which they verified off-target editing. But what they did is they not only showed the editing profile at the supersaturating dose that they tested, which you see is all the way on the right there, where you do see very, very low level. I mean, in no cases at more than 1%, and in most cases much, much less, on the order of tenths of a percentile. But you do see some off-target editing there. But the point here is they did the full dose response with those off-target sites. And what they find is that an EC90, which is much more reflective of the highest possible exposure hepatocytes would have to the drug product in a therapeutic context in the body, there's not a hint of editing at all. So this really drives home the point that, yes, if you really exaggerate your assays, if you really push them, you're almost certainly going to find something or the other. But they may have absolutely no physiological meaning, any therapeutic consequence whatsoever, because if you're actually considering the dose exposure in cells, whether it's ex vivo or in cells in vivo, then it's not something you should put too much stock in. So the last example I want to present deals with the last principle: does genome sequencing, karyotyping, optical genome mapping, and so forth demonstrate any structural variants involving the site? And if so, do we have to worry about it? And as I mentioned earlier, it's going to be most relevant to nuclease editing, although there is the possibility of these sorts of structural variant edits, off-target edits, even with other editing modalities. So this is an example from CRISPR Therapeutics, Vertex Pharmaceuticals with Exicel, better known by its brand name Casgevy. This is an ex vivo hematopoietic stem cell directed nuclease for sickle cell disease, beta thalassemia, a variant-aware off-target analysis performed by an academic group. So I need to be very clear about this. This was not actually with Exicel itself, but rather with the nuclease, and the same nuclease, the same guide RNA in the hands of an academic group that were unaffiliated with CRISPR Therapeutics. So a variant-aware off-target analysis did verify a site of off-target editing on the same chromosome as the on-target site, as shown here. Now, what's important is that this off-target editing only occurs if there is a minor allele of a naturally occurring variant that is present in about 10% of individuals of African ancestry, right? So this does not affect all patients who are being treated, but a relatively small minority. But it turns out if that particular genetic variant is present, then it affects a site that is on the same chromosome as the target gene, which is BCL11A, which is what you see on the left of this diagram towards the left. So on the short arm of chromosome 2, the off-target site that's created by this naturally occurring genetic variant is on the opposite end of the chromosome. It's on the long arm 2Q, and it's near a gene called CPS1. And so the question is, does this present any biological risk? And so it does turn out that, at least in the hands of this academic group, that in cells, hematopoietic stem cells treated, exposed to the nuclease and the guide RNA, that there was a low level of editing, And in fact, there was a low level of structural variation, inversions involving these two sites on either side of the chromosome. And so it seems likely that some patients who are receiving this drug might be experiencing this. At the advisory committee meeting of the FDA for exa-cel's approval, the full discussion that day, as I recall, almost entirely centered on this one observation. I don't think there was any other aspect of the drug that was particularly concerning to the FDA. So there's a robust discussion about this. How much risk does it present? If you think about it from first principles, if you have inversions in this chromosome, in hematopoietic stem cells that are now being engrafted in patients, any descendant cells potentially are going to have this inversion, but you won't actually have aneuploidy, right? Because the descendant cells, they're still going to get the full complement of chromosomes. They just won't be in quite the right configuration. It'd be much more concerning if the off-target site was on an entirely different chromosome and you had chromosomal translocations, because then the descendant cells of those hematopoietic stem cells would not have the full complement of 46 chromosomes, or the normal complement of 46 chromosomes. They would have extra material or missing material, and that might be much more concerning with respect to oncogenic risk. That's inversion. Now, if you're actually talking about potential disruption of the CPS1 gene itself, it's not a cosmic gene. There's no particular reason to think that having a low level of indels locally affecting CPS1 function would be particularly concerning. So that's really what I want to do. I just want to present some cases to illustrate these various principles. I'm showing them again on this slide. I think something that we could consider and we should consider is this idea that instead of generating a long list of nominated sites using various techniques that were outlined nicely by Dr. Naldini, and this is what we are all doing. We're using at least 2 orthogonal techniques, generating these long lists of candidate sites, and then making a serious effort to actually do verification of that long list of sites and look to see if there's any off-target editing. You could very well ask the question, well, if you do find off-target editing and you know a priori that there's going to be a very, very low risk of a biological effect going through a framework like the one that I've shown you in the slide, you could very well ask the question, well, why are we even taking the time to verify those sites? Because even if we found something, if we're already reassured that they don't present biological risk, then there isn't much point in doing the empirical testing, right? So what I would suggest is we could start to think about bioinformatically applying this framework to candidate sites that have been identified through the initial screening process. And then if none of those sites or few of those sites actually from this bioinformatic approach present risk, then perhaps put them aside, not go through the serious exercise of doing the verification, because even if you found something, it's not something you would be concerned about. I know that's a little bit out there, but I think where this becomes very important is if you start to think about platformization and you start to think about patients with significant unmet medical need, where the time available to do this sort of analysis is very, very limited, then you're going to have to do things in an expedited way. There's not the luxury of spending months to years and lavish amount of resources in doing the best possible off-target analysis. And so I would suggest that you could apply the bioinformatic framework, eliminate all those sites where there's not any serious concern of biological risk, and then focus the verification phase on just that small number of sites where there might be concern, and then hopefully rule them out. And if you don't rule them out, then you have to ask questions whether you want to pursue the program or not. So that's really all I have to say. I think I've exhausted the time available to me, and I think you can either ask me questions now or we can launch directly into the Q&A portion of this presentation. Thank you very much.
Many thanks, Dr. Musunuru. Quick questions to this presentation. So, Alessandra.
Yes, thank you, Dr. Musunuru, for sharing these 3 very illuminating examples. I have a comment and a question. The comment is that the example of Wilson disease, ATP7, tell us that not need to scare, and tell us also to take into account the the knowledge of the autosomal recessive diseases, so meaning that you are disrupting an allele, that doesn't mean that you have a medical effect. And the question is on the first case of the PTEN. PTEN story is very interesting story. So I'm wondering if you thought to use this cellular model in order to moving forward and to understand if having this off target on the important cancer gene like PTEN is this sufficient to develop cancer, for example? Because this could be a very useful model in order to demonstrate that even though you have the mutation, not necessarily you develop a cancer. For example, in culture cells.
Yeah, we gave serious thought to that. And what we realized was we could go through that exercise, and from an academic perspective, it could be very interesting to see how well we could model that sort of effect in, as you suggest, the cell cancer model, or even in vivo. We even considered that. But the ultimate goal was to develop a drug that would benefit patients. And in this case, we actually had different guide RNA we could use to achieve the same effect. And when we very exhaustively announced that other guide RNA, there was no issue whatsoever. There was nothing pointing to the same gene, the same site, because it was a different guide RNA. And so we felt the better part of valor was to simply shift our effort to the other guide RNA rather than going through this, extensive exercise of biologically de-risking a guide RNA that ultimately we weren't going to use. Now, if our options had not permitted us to pivot to another guide RNA, then I think we probably would have embarked on exactly the exercise. So model it in cells in a dish. It would have to be some sort of immortalized cell line, which is non-optimal, but you can't really do much with primary human hepatocytes in the dish. But the idea is to engineer that particular off-target editin, see if that had a biological effect, see if cells with that particular variant were becoming more prevalent in the population, suggesting that there was a positive selective advantage to having that variant. And then more broadly, asking the question of whether you gave a high dose of that base editor to the cells in the dish, whether you would actually pick up structural variation affecting the gene and potentially disrupting the gene, which obviously would be much more concerning because there you would be directly, presumably, inactivating a tumor suppressor gene. But fortunately, we had other alternatives, so we did not have to go down that road.
Thank you. So, Tineke?
Yeah, so I have also a question, maybe a bit of a detailed one. Thank you for your presentation. So with regard to the dose dependency in vitro versus clinical, with the non-clinical research, we call it often a safety margin. So do you only need to take into account the dose, or would also a factor such as time maybe be different, like in a Petri dish versus clinical, where the guide may be more easily be broken down, or when you apply the enzyme via an AAV or directly as a protein?
Yeah, I think that's a very important consideration. So in the drugs we've been developing have been largely lipid nanoparticle drugs. And so the idea there's very short-term exposure to the drug because the messenger RNA drug substance, the guide RNA drug substance, they fairly quickly break down within the body, within the cells. The exposure is a limited time frame. It's going to be an entirely different scenario if you're using something like a viral vector, like an associated virus AAV, as you mentioned, or a lentiviral vector, where there may be more prolonged expression of the editor. And that's going to be more challenging to model ex vivo. That's not to say you can't model it and should model it. But in our case, what we have been doing is we're using our lipid nanoparticles. Obviously, the goal is to treat cells inside the body. We use the same exact drug product that's intended for human patients and treat the cells outside the body, with the reasoning that that's probably a reasonably conservative approach to doing it, rather than doing something else like using a viral vector with lenti and constitutively express that for a very long period of time, or using even plasmid-based transfection, which would present much higher exposure to the guide RNA and the editor than using a lipid nanoparticle delivery of the messenger RNA to guide RNA.
Thank you.
Okay, so now let me open the floor to all the participants for the Q&A session. So please raise your hand and then we will unmute you. We will not have time to look at all the comments you put in the comment box, but of course we will not ignore them. We will look at them later and see if there's anything that needs our input or our reflection afterwards. See that, Fyodor, you raised your hand. Feel free to take the floor.
First of all, thank you, Drs. Naldini and Musunuru, for just setting the stage so magnificently. I want to ask the agency to think forward to 2030, when there are, I hope, approved medicines for CRISPR on demand, and specifically in severe disease, and think about the concept of pharmacovigilance. Skysona is a great example to me, right? It's a horrific disease, just absolutely horrific. And it's on the market. And it's obviously, I forget the latest numbers. The numbers I have is, I think about 6 patients treated so far have developed treatment-related iatrogenic malignancy because of insertional mutagenesis by the vector. The product is on the market because the benefit risk is commensurate with the disease indication. So forward integrating to 6 years from now, when there is a platform for on-demand treatment for devastating inborn errors of immunity or other diseases, where people get their own CRISPR and there is a nominated set of off-target sites, and a fraction of the subjects on the trial develop some iatrogenic event. We all hope this doesn't happen. But I think it's clear that personalized guides will have personal off-targets, as Dr. Naldini pointed out. And the question is, what happens if you have a guide that works really well but has an off-target in PTEN? I really think we need to start thinking about what pharmacovigilance is going to look like, because I think in a non-clinical development program that Dr. Musunuru described, if you can just switch to a different guide, let's go. That's exactly what to do. But in an approved medicine where you know that there is the potential risk, I think thinking hard about benefit risk considerations, whether one withholds the treatment from the patient because of this hypothetical but non-zero risk, or alternatively, whether one establishes a pharmacovigilance program where one monitors potential iatrogenic effects over time and administers the therapy to the subject because the alternative is severe disability or death. I would just like to put this up for consideration as we think forward to a world where CRISPR on demand is commercial reality.
Okay, I have Luigi. Please go ahead.
Well, I second again Dr. Urno's point, and I think just as a follow-up to his mention of the leukodystrophy story, I think the key problem for that, of course, is that the specific vector design that was used, but we knew that in advance. We knew because there was clonal monitoring of patients. And we did, and they reported, and we pointed out the risk because there were clonal expansion in those patients years before those leukemia developed. But the company was reluctant to change that vector, although they did acknowledge the specific risk because of the cost of the burden to not go back to an improved version. So that's again called for this issue of regulatory time and cost of development. That's another very important point. We are all starting clinical trials with vector, reagents, editor design, which are not perfect by definition, but they are not also the ultimate one. And while we are testing in the clinic, we maybe develop a better version. There should be a better and easier track to improve and upgrade those versions, even when they have been approved. We have to realize companies are very reluctant to do that. And again, we should push that better. The second point I want to go is after Dr. Musunuru point, I think I'd like to take the challenge you mentioned. Can we bypass some of the verification and validation thing because of those very reasonable arguments that he made, because we can anticipate risk or lack of risk because of target. So there's 2 points that I like to make. One is that an off-target may be meaningless as a specific site of changes, but it may become a site of a translocation if it is your editor is not producing breaks, as I think you mentioned. So maybe we should couple that strategy, say, let's forget about deep validation, but then let's rely on some genome-wide assessment, whether there is a significantly evidence of unbiased, by an unbiased method of translocation or aberration. So that's maybe good enough to potentially validate this approach to bypass some of this deep analysis. What about that?
Thank you. Any questions from the audience? Fyodor?
I think that the real application of artificial intelligence should be not in recording what we say, but automatically and muting when somebody is trying to say something. That would be a great AI algorithm. Sorry about that. So Alexis Cockcroft says, is it feasible to aim for a globally approved algorithm of best practice to decide upon an acceptable candidate? I think this is an essential goal. And the only way to do that is to pool clinical data. And that's not simple to do. But I think, especially for rare patient populations, I think that having regulators get together, there are existing frameworks in which the agencies work together, but like learning from clinical evidence and then deciding what matters and what doesn't, I think would be key. And one of them, again, as I mentioned, pharmacovigilance, certainly for hematologic disease indications. And, a really important example is this very famous subject on a European trial for gene transfer thalassemia. The subject became oligoclonal but then that oligoclonality resolved. So I think we just need to think forward to a time 5 years from now when we can look back and use these clinical data to synthesize these more harmonized best practices. This doesn't mean that we shouldn't sort of try to do this now, but I'm going to argue that specifically for gene editing on demand, it's kind of early days. So I think data sharing and benefit risk considerations will be key, but I really second Alexis's motion.
Yes, a brief comment from my part. I think as a regulator, one of the issues is going to be, because we are talking about platforms, but we're also talking about different benefit risk. And so I think the comfort of the regulators also very much depends on how narrow, in a way, the platform is, because we get the presentation where you move from a highly debilitating, life-threatening disease and you call it a platform, which includes not so life-threatening diseases. So could there also be a discussion of how reasonably wide that platform concept could be to meaningfully do leverage the clinical data in a way? And while there could be an expansion at a later stage, maybe, can there be a discussion of how limited should it be in the first place to really have meaningful data that we can analyze? Any further comments, questions?
Rune?
Yeah, I'm just a bit curious as a regulator. I don't know what practical experience you had with regulators. I guess it's mostly from the FDA, from your side, but maybe you had some interaction also with the EMA. I don't know. So what's your impression? I mean, this is a learning curve for all of us. And of course, I think it's in the nature of regulators to be a bit conservative. So far, are we sort of off target in our demands when it comes to what we demand on non-clinical quality, documentation, etc.? What's your impression? I think it's helpful for us also to get some feedback, and because we need to learn also.
Yeah, so I will just say, look, I mean, first things first. Rule number one in cell and gene therapy: don't work against the agency, work with the agency. My lived experience, again, this is Sangamo and then biotech companies I'm connected with, and now my work at the Innovative Genomics Institute, is the agencies, certainly I've worked most with the FDA, have been consistently reasonable and engaging in a dialogue, and I'm very grateful for that. My one request to the agencies moving forward is certainly the EMA. It was very encouraging to hear that there are ways to, expand the scope of a trial, is to think vigorously with us about what it means to de-risk a gene editor that will be used once, such as Rebecca Arntz-Nicholas's clinical trial for one child with CPS1 deficiency, versus de-risk a gene editor that is going to be used on 1,000 people, such as Intellia's medicines. I'm not suggesting we treat patients with dirty guides. That's not what I'm suggesting. But I think that just making sure that the regulatory review and regulatory considerations on the path of a given editor to the clinic are consistently made commensurate with two things: the unmet medical need and the standard of care. I mentioned etoposide, which is administered to children with pediatric HLH, as one example of a severe, sort of quite remarkable. And then separately, how many times this drug is going to be used. I think that this is a challenging predicament because this is going to be disease by disease specific. And even in inborn areas of immunity, where much of my life happens right now, some of these have treatment options, some of these don't, other than, haploid integral bone marrow transplant. But my one request to the agency is consistent focus on benefit risk, size of patient population, and making sure that the regulatory framework is commensurate with those things, rather than slightly more, across the board, no matter the size of the disease indication and no matter the disease severity. That would be my request.
And so sorry, I apologize for having to step away for a moment there, but my perspective on this is informed by the time I spent working with Verve Therapeutics, who have been developing gene editing therapies for hypercholesterolemia, atherosclerotic cardiovascular disease. So they were perhaps the first, if I remember correctly, to actually go to the FDA with an IND application for an in vivo gene editing therapy. If not the first, then maybe one of the first two. And the FDA acted very, very conservatively, to the extent that the drug product, the program, was placed on a clinical hold for 10 months and was almost entirely about further assessing the possibility of off-target editing. At the start, before the clinical hold came down, there was no evidence of off-target editing by the guide being used in that drug product in the target tissue, hepatocytes. At the end of that 10 months, which represented a huge amount of work on the part of the folks at Verve Therapeutics and untold resources being spent on a whole variety of different identification assays, verification assays, and so forth, not just the liver, but many, many tissues across the body, there was still no evidence of off-target editing. And so clearly that was way too much energy spent on assessing for something that didn't turn out to actually have any meaning at all. Perhaps understandable because it was early days. It was one of the very first gene editing applications. None of us really knew the best way to go forward. But I think that just illustrates how being very, very conservative can be highly detrimental to a drug development process, because that delay of almost a year means an extra year that patients who could benefit from the therapy are not able to access that therapy. That's my sense of it. Now, I do know the FDA has changed their thinking dramatically in the several years since then, and that similar programs that are now being considered with IND applications are not having to go through anywhere near the same amount of scientific scrutiny, in the sense of having to do these exhaustive analyses as Verve had to do there back at the beginning in 2021, 2022.
Thank you. I think this is, of course, being the first one coming with a novel approach, always is paying a lot of money or a lot of effort that the second and the third one will benefit from. That's, I'm afraid, the case, but it's good to remind this. And you mentioned this now for gene editing, but we have seen this for the first gene therapies, AAV-based, exactly the same with the first cell-based products. Yeah, okay. Professor Naldini?
Well, that was exactly my point, I guess. So, and again, I don't have personal experience of discussing genome editing with the regulators yet. As I said, we will start engaging this very soon, hopefully, but I have probably 20 years of interaction with both the EMA and our interaction, even some FDA with the lentiviral technology. I fully understand, as you just said, Patrick, the reason to the caution and the need for extensive initial data when the new technology enters clinical testing, and that makes sense. What I think, in my view, I have not seen is, compared to what I hoped, was a significant mitigation of those requests and approach as we move forward and we accumulate years and years of clinical experience, which overall are substantially safe. I think that effort, which is composed in part of the concept of platform approach, but also in part on the capacity really to exploit existing data, which should not be necessarily included into an application because they are common knowledge, they are available, they belong to the platform. I think this type of effort is, I think, could be more. There could be more effort on that side because we have learned through the clinical experience what we could not prove initially. And I think, as you just said, a technology which is novel versus a technology which has been fully validated for a decade should be very differently approached.
Yes, I do hope that with events like these, we are moving in the right direction on these issues. For me, as a quality person, there are two issues. You've talked about optimization. I think there needs to be a discussion about how can you demonstrate that it is indeed an optimization and not an entirely different product. And I think this is where experience needs to be leveraged and where we need to learn. And the other significant issue for me is not a direct manufacturing issue. You were talking about of the N=1 treatments, and there are legal exemptions for these kinds of setups. However, the data generated and the requirements are very different compared to what we would normally require for marketing authorization. So the balance between finding an approach that allows the generation of high quality data that will allow us to learn and indeed see the similarities and commonalities, but at the same time enable fast access within the regulatory framework. I think that is the challenge for moving forward. In other words, how much is really truly different and how much is truly common to the approaches that we are faced with. And I think as regulators, this is one of the areas where we need to benefit from your experience in order to have the flexible approach and just judge the benefit risk of a given approach. Thanks.
Okay, Kieran, you have raised your hand still. You want to contribute?
Yeah, if I could address that comment that was just made, which I think was actually a very wise comment. So I related the first part of my experience interacting with the FDA with respect to Verve and a therapy that is ultimately intended to be used by many, many, many patients for a very, very common indication, hypercholesterolemia, atherosclerotic cardiovascular disease. But the contrast to that was our recent experience with the drug KA that we made for baby KJ. And Dr. Ahrens-Nicklas and I will speak more to this in the afternoon. But the short version is because time was of the essence, we didn't have the luxury of months and months or years of time to do a comprehensive off-target analysis. The FDA took what I thought was a very enlightened view, which was, do the identification assays, come up with your list of nominated sites, prioritize those sites you think are of the highest concern, and it ended up just being 24 sites out of the list of potentially hundreds of sites. De-risk those, present those data in the initial IND, in this case, a single patient expanded access IND application. In the background, as things are going on, continue to do work, continue to qualify all those additional nominated sites you took. It won't actually affect the treatment. They gave us the go-ahead to treat KJ with that limited amount of off-target analysis, but they did want to see as an appendix to the IND application months later, a more robust analysis of the off-target. So I think it was a nice balance of, not standing in the way of getting a patient a very, badly needed treatment, but still gaining important scientific and regulatory knowledge out of the experience.
And I think that's a nice conclusion as well of this Q&A. We expanded a little bit the time, but it was certainly helpful. We close here the morning session. I thank all the presenters and the experts here in the room as well for their contributions and input. And we see each other at 1:10. Welcome to everyone. So we have a quite packed afternoon session, so we will start immediately. A presentation from Dr. Musamuro and Dr. Arends Niklas.
Thank you. It's a real pleasure and privilege for both of us, Becca and I, to have the chance to speak to you today. You can see our various disclosures at the bottom of the slide. I should note that our specific instructions were to identify scientific challenges of in vivo and in vitro gene editing medicines. Rather than speaking to platform technology approaches per se, we have plenty to say about platforms, but we'll put that aside, ignore the title we have for this presentation. For today's purposes, we're going to largely focus on the scientific challenges we faced and had to overcome in developing an N of 1 therapy for a specific patient. In late summer 2024, not a little more than a year ago, a baby named KJ was born. And on day of life 2, he became very ill. His ammonia was found to be greater than 1,000, and he was rapidly started on life-saving dialysis to remove this neurotoxic ammonia. By the time he was a week old, he was diagnosed with biallelic nonsense variants in the CPS1 gene. And unfortunately, this meant he had the most severe form of the most severe urea cycle disorder. This condition has a very, very high infant mortality, greater than 50%. We noted that one of his variants that were identified by genetic testing that was done at CHOP within several days of his birth showed that he had the Q335X variant in CPS1, which was potentially amenable to adenine base editing. After KJ was stabilized, he was started on a protein-restricted diet, ammonia scavenger medications, and citrulline supplementation, which is all the standard of care. He remained admitted to the hospital while awaiting a definitive therapy, namely a liver transplant, which typically he wasn't in a position to get until he was at least 1 year of age in order to be able to get a liver transplant safely. So our first step in trying to develop a gene editing therapy for KJ was to devise a way to very, very quickly figure out a base editing solution to the CPS1 Q335X variant in hepatocytes. So we generated a lentivirus transduced HUH7 cell line with 4 variants, the patient's 2 CPS1 variants, which you see there, as well as 2 reference control variants in the PAH gene, which cause an entirely different disease, phenylketonuria, or PKU. This patient-specific cell line was ready before KJ was a month old. So why did we use a lentivirus transduced cell line? It was all about speed. We realized that we couldn't let perfect be the enemy of good. It would have been nice to be able to introduce the Q335X variant directly into the endogenous CPS1 locus, but it just wasn't going to be possible to do so in a short time. We recognized that there was no particular need for the variant to be in the endogenous locus. It just needed to be somewhere in the genome. And that's because absolute editing levels were not so important. What we really need to know were the relative editing efficiencies of different editor guide RNA combinations that we would screen and which specific combination performed the best. In terms of various options of quickly inserting sequences in the genome, one could use lentivirus, a transposase, or an integrase. They'd all be fine, but lentivirus has the advantage of being relatively accessible to most research laboratories. Once we had the cell line, we could use it not only for screening for the optimal editing solution, but we could also use that same cell line for a potency assessment for the final drug product, which you'll see later in the presentation. However, in all of this, an important consideration is that we felt we needed to include at least one variant that could serve as a well-validated positive reference control. In our case, we used 2 such controls, the 2 PKU variants. Now, why did we use the 2 PKU variants as controls? Well, it's because we had prior data establishing a robust in vitro, in vivo correlation. We had shown that lipid nanoparticle or LNP test articles with an adenine base editor mRNA, and in each case, the optimal guide RNA could achieve rather high levels of in vivo corrective editing of the variants in the liver in humanized PKU mice with the variants. and fully correct the elevated phenylalanine levels in the mice, the hallmark of PKU, within just 48 hours. And you can see the orange curves in each of the graphs. So our thinking was that if we could find a corrective editing solution for the patient's CPS1 Q335X variant that matched the editing efficiency of one or both of the PKU control variants in the same cells, in the lentivirus transduced HUH7 cells that have all the variants, that would be a good sign that the corrective editing solution might work well in vivo, specifically in our patient, in KJ. So we used the lentivirus transduced cell line to screen candidate editor guide RNA combinations for correction of the patient's Q335X variant. I should note that all of the bystander editing at this site results in synonymous variants, which should not affect the function of the CPS1 protein. The best editor was SPGABE8E, which has a relaxed PAM preference for NGN. We had this solution in hand just 4 weeks after KJ was born. Now, we did think it'd be worth spending a little extra time optimizing further, so we reached out to Ben Kleinstiver at Mass General Brigham to see whether he had any novel editors with novel PAMs that could better match the extended NGCC PAM for our winning guide RNA. And he sent us 3 candidates, and they all looked good, surpassing the previous winner, as well as the PKU reference control that you see on the left-hand side of the graph. Now, on the advice of David Liu at the Broad Institute, we then installed the V106W variant into the deaminase domains of the editing proteins to reduce off-target editing and potential toxicity. And we had a clear winner highlighted with the red box. We refer to this editor as NGCABE8E V106W. We had this done by 6 weeks after the patient's birth. Once we reached this point, we reached out to academic and industry partners to help us figure out how to actually manufacture the drug and make sure it was safe. So our partners included Fyodor Ernov and his colleagues at the Innovative Genomics Institute at UC Berkeley. You heard from him earlier. Integrated DNA Technologies, IDT, manufactured the guide RNA drug substance. Aldebaran manufactured the messenger RNA drug substance. And Acuitas Therapeutics contributed their LNP technology, working closely with Aldebaran to formulate the LNP drug product. IDT and Aldebaran are subsidiaries of Danaher Corporation, who provided oversight of the entire manufacturing process. And you'll hear all about that from Sadik and Vanessa from Danaher later in this afternoon session. Now, in parallel with all this, we'd already started the process of generating patient-specific mice in which to test the optimized base editing solution, because we felt that at least the first time we did this, it was important to generate some in vivo data. So we generated a multivariant mouse model with the same 4 variants we put into the cells, KJ's2 variants, and then the 2 control PKU variants. So that same cassette, we inserted it into the ROSA 26 safe harbor locus. And the advantage of doing it this way is that we could use well-validated CRISPR reagents targeting this locus in mouse embryos to very quickly and 100% reliably make the mice. And indeed, we had 2 founder mice by 2 months after KJ's birth, and we were able to breed them and obtain clean F1 heterozygous mice a couple months after that. Now, we did also attempt to make a standard knock-in mouse with the Q335X variant in the endogenous CPS1 locus in the mouse genome, but that was much more challenging. We didn't get mice in time to do anything meaningful for this project. So about 4 months after KJ's birth, we had a pre-IND meeting with the FDA to ensure that our proposed plan of IND-enabling studies would support the safety of the therapy we were developing. They were incredibly supportive of our effort, and they committed to a rapid review of our IND application when we ultimately submitted it. So using the winning base editor guide RNA combination, IDT, Aldebaran, and Acuitas completed a toxicology batch of the drug, which ended up being named Kjageran Abangseveran, or K-ABE for short. We had this drug in hand a little more than 4 months after KJ's birth. And using the tox batch of the drug, we performed a small non-human primate toxicology study, which showed that a very high dose of the drug was well tolerated, with the only findings being transient asymptomatic hepatic transaminase elevations, which have been reported with other similar LNP therapies. We also found that the lipid components of the LNP drug had almost entirely cleared out of the bloodstream by 2 weeks after treatment, and this gave us the confidence to propose to the FDA a repeat dosing protocol for the drug with up to 3 consecutive doses separated by a minimum of 3 weeks each. This study was completed by 5 months after KJ's birth. Now, by this point, we had in hand 6 F1 heterozygous ROSA 26 multivariant mice, the design I showed you earlier, that we could use to test the toxicology batch of the drug for editing efficacy in vivo. We performed a small dose-response study with the drug and observed up to 42% whole liver corrective editing. Keep in mind that here the bystander editing is all synonymous variants. We saw editing even with the lowest dose, 0.1 mg per kg, which was our proposed starting dose for the clinical protocol. We completed this work when KJ was 5 months old. IDT, Aldevron, and Acuitas completed the clinical batch of the drug, the same drug that would be used to treat KJ a little less than 6 months after KJ's birth. We tested the drug in our lentivirus transduced HUH7 cell line, doing a potency assessment, and we saw nice dose-dependent editing with some bystander editing. Again, we were lucky that for this variant, virtually all bystander editing resulted in synonymous changes that wouldn't affect the protein function. The calculated EC90 was about 50 nanograms per mil in terms of the amount of drug in the media, and so we decided to go 20-fold higher, a dose of 1,000 nanograms per milliliter as a supersaturating dose with which to perform off-target analyses. So working with Theodore Ernoff and other collaborators at the Innovative Genomics Institute, as well as at IDT, we performed the off-target analyses with the clinical batch of the drug. We performed an extensive off-target analysis in a relatively short time. We used 3 orthogonal techniques, OneSeq, developed by Keith Jung's group, who were at Mass General Brigham at the time. It uses a synthetic library of oligonucleotide sequences chosen from a reference genome for their similarity to the on-target site, nominating a list of sites that are base edited when exposed to editor and guide RNA in the test tube. ChangeSeq BE, developed by Shendar Sai's group at St. Jude's Children's Research Hospital, uses genomic DNA isolated from cells, again, nominating a list of sites that are base edited when exposed to editor and guide RNA in the test tube. Our collaborators at the Innovative Genomics Institute performed ChangeSeq BE for us. UncoverSeq is a version of GuideSeq that's been developed by IDT. It's a cell-based assay and nominates yet another list of sites. Now, because we were focused on one very specific patient, KJ, we realized that we had a great opportunity to do a patient-specific off-target analysis. So for OneSeq, we were able to use KJ's own genome as the reference genome since he had undergone whole genome sequencing at CHOP as part of his genetic testing when he was born. For ChangeSeq BE, we have tried to obtain enough genomic DNA from KJ. We weren't able to do so. So instead, we went to KJ's father, who shares the Q335X variant, and obtained genomic DNA from his blood samples. Now, after we performed the 3 techniques, we integrated the 3 lists of nominated sites and selected the highest priority sites, those that were top ranked or that were located in gene exons. And we used individual targeted amplicon sequencing to interrogate these candidate sites in our lentivirus transduced HUH7 cells, as well as primary human hepatocytes from 3 infant donors using cells that were either treated with a supersaturating amount of LNPs or were untreated. And we observed the expected corrective editing of the lentiviral Q335X variant in the HUH7 cells, so that was good. We also observed low-level bystander editing at the wild-type, on-target, endogenous CPS1 site in all of the cells. That makes sense because the guide RNA has only one mismatch to the wild-type site. We observed only minimal off-target editing in HUH7 cells only in a deep intronic site in the ATP7B gene. I mentioned this in my talk this morning, encodes a copper transporter and was not felt to present oncogenic risk. All this work was completed 6 months after KJ's birth. We subsequently interrogated hundreds of additional candidate sites with IDT's RAMP-seq, a multiplex PCR method, and didn't identify any additional off-target sites, which we ultimately reported to the FDA several months later as an amendment to the initial IND application, although we didn't let that hold up KJ's treatment. So finally, 6.5 months after KJ was born, we submitted our single patient expanded access IND application to the FDA, and the FDA approved the application in exactly 1 week. I'm going to turn it over to Becca now to tell you what happened next.
Great. Thanks, Kieran. You can go forward a slide. And so, as Kieran said, right before he turned 7 months old, we received permission from the FDA to go forward with dosing. And he received ultimately 3 doses of the medication. And we'll talk about what happened. So one of the challenges when you think about advancing these types of therapies is how do you choose a starting dose for an individual patient? One of the benefits of LNP base editing therapies is that they are able to be redosed. Prior to our report, there had been reports of adult patients, very few, being redosed with base editing drugs, but we wanted to see if it was possible for an infant. And so when I designed the clinical trial or the clinical care guidelines for this case, we designed a multi-dose ascending dose protocol for dosing. And so as you can see here, the doses he actually received started at a very low dose of 0.1 mg per kg, went up to 0.3, and then 0.45 at the intervals shown. I should say that dosing schedule was decided upon by an oversight committee, so it was not all decided by us, but rather a group of physicians, including immunologists, metabolic physicians, gene therapy experts, our liver transplant team, that would review all the data between each dose, both looking for signs of safety and efficacy, and then collectively we decided whether or not another dose was indicated. Luckily, he tolerated the medication very well with no serious adverse events. He had a brief coughing episode at the beginning of dose 2 and 3 that were self-resolved with just repositioning, and he was able to tolerate the full infusion at the full rate with no other effects. He had a low-grade fever and transient rash after dose 3. And then we did see the mild transient asymptomatic increases in liver enzyme levels with no other liver function abnormalities. So what you're looking at here, those dotted lines represent the dosing days. And as you can see, especially after dose 3, you see a sharp increase in his ALT that resolved with no sequelae and no treatment. Those gray bars show episodes of viral illnesses, which he also caught during the peri-dosing period, which also increased his liver function enzymes, which we see on a regular basis in our pediatric population. Now, everybody wants to know, did this work? I'm going to say flat out, this is a N of 1 clinical experience, right? This was an expanded access, single patient investigational new drug application. Technically, it occurred as clinical care. It is not a clinical trial at this point. But we can report what we observed. And what we observed is that very shortly after that first dose, we were able to increase the amount of protein he could tolerate in his diet. And that's what the first panel is showing. Prior to dosing, he could tolerate about 70% of the recommended daily allowance for protein intake. And after that, and since his first dose, we've been able to increase to actually a little bit above 100% of his recommended daily allowance. We were also able to wean some of the medications he requires, his nitrogen scavenger medications. And in doing all of this, he maintained normal biomarker levels, including his glutamine and ammonia levels. And he had a really nice growth spurt. He continues to grow very well to this day. Since dosing, the other thing we have been measuring is his blood urea nitrogen level. BUN, or blood urea nitrogen, obviously is made in the urea cycle. And what we can see is that especially after dose 3, we saw a really nice increase in this biomarker. Next slide, Carrie. So what have we learned from this experience? I think it's really important to say, again, this was a single patient expanded access IND. Making broad claims about efficacy is hard. We're going to need formal clinical trials. But what we can say about KJ is that he will likely continue to need some urea cycle management. But early signs, and I will say that this continues to be our interpretation, suggest that his disease is less severe. He's home. He was discharged from the hospital. He's doing quite well. But hopefully this also shows that it's possible to develop a personalized gene editing therapy in 6 months, despite all the challenges. And there were many. And this process was definitely met with a lot of roadblocks that we had to figure out our way to work around. We also hope this shows that repeated doses of an LNP base editing therapy can safely be given to an infant. But we really need both longer follow-up studies of non-invasive markers of editing. That is one of the challenges of this type of therapy. We do not have results from a liver biopsy for KJ. It was felt by our ethics team, and in all honesty, I agree with them, that the risk of the procedure of a liver biopsy in an infant outrisks the possible benefits. So one of the challenges is to develop better ways of measuring how well editing is occurring after one of these medications is administered. But ultimately, we need to move from N of 1 studies, these clinical care experiences, to platform clinical trials if we're going to make definitive statements about safety and efficacy. So how do we do that? What type of way, how can we streamline this process to get the definitive data that we need to decide if these are the right types of therapies to move forward for our patients? One thought that we have talked about a lot is going forward, if we use the same lipid nanoparticle formulation and adenine base editors to correct a pathogenic variant to wild type, and all we do is change out that guide RNA telling the editor where to go, are additional non-human primate studies necessary? Are additional animal toxicology study necessary at all? Are any animal studies necessary? We really think that the toxicity of the drug is dictated by that lipid nanoparticle. itself, and that will be shared between individual subjects. And the off-target risk, right, where that guide RNA might go in the genome, is really dependent on the guide RNA and should be studied in human cells, not animal models, because that's the genome that we care about in this case. And so are animal studies really necessary? So the question we have been asking is, when would cellular studies alone, both for the on-target and off-target assessment, be sufficient? And when does the severity and unmet need of the disease create a favorable balance of risk and benefit? And so this is just showing we could expand this very quickly. We have screened a number of base editors for correcting other urea cycle disorder variants. There is no shortage of need in my patient population and the patient populations with patients with urea cycle defects around the world. And if you use the approach that Kieran outlined earlier in the talk, you really can, for a number of variants, not all variants, obviously, not everything is amenable to adenine base editing, but for a significant percentage of variants, you can find a solution. The question is, how do you scale up that solution in order to actually get it into a patient? And so this is just showing you data from a number of different variants, including other CPS1 variants, other urea cycle disorders, including ASL or ASA lyase deficiency. So this really can scale quickly. So how do you meet the needs of all these patients that we might have editing solutions for? So what we are proposing and what we are really trying to advance is a master protocol for both urea cycle disorders and the related disorders, organic acidemias, where clinically the benefit of liver correction is very clear in that patient population. And so what we have been proposing is that you have a master protocol, IND, for severe neonatal onset disorders of both urea cycles and organic acidemias. And then you have a primary IND, and if you have a new patient that is born or a new patient that presents to care that has a variant that is amenable to a particular base editor, we are asking permission to essentially add that variant into an existing investigational new drug application for that particular disorder. And then the efficacy and the safety of those variant-specific versions of the drug product would be compared essentially in umbrella trials specific to each group of disorders, whether it be urea cycle disorders or organic acidemias. And we are very lucky that this work has been heavily supported by the Somatic Cell Genome Editing Consortium of the NIH. Kieran and I are actually both at a meeting of this group this week. And the amazing opportunities that this network has provided is not only just funding opportunities, but building that network of collaborators that Kieran mentioned, and building a network of collaboration with our regulatory partners to really try to advance these therapies in a thoughtful but efficient way. And I think that is one of the biggest things we have learned about how to overcome challenges in this space, is that you have to collaborate with others and you have to work together, because not one person can do every step of this process. So I think building those networks and building those collaborations are going to be key to overcoming the challenges of developing personalized therapies. And with that, speaking of collaborations, you can see from this slide, this is not an all-inclusive list of everyone who helped contribute to this project. It really has been a massive team effort. And first and foremost, I want to thank KJ and his parents, because they really put their trust in us, and were the best collaborators and really joined the team very early to help expand this work forward. And with that, I think we're happy to take questions or answer anything that people may have.
Okay, many thanks for the very interesting presentation. We have a couple of minutes for questions. So looking first in the room here, are there any direct questions or comments to the proposal made? Marcos, yeah.
Yeah, thank you very much for the very nice presentation. I have a comment/question. The first thing is that it is amazing what you did, but it shows that it can be done. So, and you did it hand by hand with the regulator. So that shows that it is possible to do it. Of course, it's impossible to think that this level of work can be done for every patient. But my question is, when you talk about this umbrella development, are you thinking of all urea cell cycle disorder, or just based on specific genes, for example, or what do you think will be more feasible at the moment? Thank you.
Yeah, I'm happy to answer that one. Yes, we are thinking of all severe urea cycle disorders, right? So not every patient that has a urea cycle disorder presents in the neonatal period, But the patients with the greatest unmet need are really our severe urea cycle disorder patients. And there's 7 genes that tend to present this way. And so if a subject is identified, or if a patient is identified to have a pathogenic variant in one of those 7 genes that's amenable to this type of base editor, and that we can measure efficacy down the road using the same markers, because regardless of the urea cycle disorder, we know how to measure efficacy, and it's shared across gene-specific urea cycle disorders. That is our intention, is to essentially have one master protocol, and whether you have OTC deficiency or whether you have ASA lyase deficiency, if you can measure efficacy using the same markers, such as increase in protein intake and reduction in nitrogen scavenger medicines, you should be able to evaluate it under a single investigational new drug application. And that is what we've proposed.
I have a question with regard to the repeated administration. So if you can have with a base editor also unintended on-target indels, for instance, or modifications, would you increase the risk for that if you do more treatments than one?
Yeah, so maybe I can address that. I think there is a possibility, particularly as you refer to, unintended consequences at the on-target site. But, again, the disease is the appropriate context in which to consider those risks. So these are recessive disorders. And if a patient has an infantile or even a neonatal onset urea cycle disorder, it means they basically have no function of the enzyme that's affected or the transporter that's affected, right? So it is complete loss of function. And so our view was that even if there is some level, even if it's a low level, some level of indels occurring, that would represent deleterious mutations. It's on a background of genes that already have no function. And so the goal with the repeat dosing was really try to ramp up to the maximum level we could while still maintaining overall safety, the corrected alleles that would start producing functional enzyme. And another advantage of these diseases is you don't need to correct all the alleles. You don't need to correct 50% of the alleles because these are recessive diseases. In all of these cases, we think 10 to 15% of the alleles being corrected would be sufficient.
Okay, thank you.
Okay, is there anyone from the audience who wants to put a question forward?
Thank you so much. Wonderful work and very inspiring. Concerning the pipeline of the umbrella of the platform, as you said, since these are recessive disorder, it's enough to correct just one mutation. But what is going on if none of the mutations are approachable by base editor? That could happen. So are you considering to switch to others?
Yes. In the end, we're going to need solutions that could address a variety of, obviously, genetic variants. And there are many genetic variants that are not amenable to base editing. So in the future, we would love to move to a platform such as prime editing. Technically, that is a little bit more challenging, but we're working on it. In fact, one of KJ's other variant is amenable to prime editing, and we've worked in parallel on that variant while we were trying to develop the base editing solution.
Again, just a short comment from my part. I think in principle, there is nothing that speaks against having a master protocol from a legal regulatory point of view. I think there are lots of questions on details, so it would definitely be a brilliant idea to have intense exchange, particularly with the clinical trial community in Europe, but also obviously for the later marketing authorization. But just to say, there's nothing that principally speaks against the concept also being used in Europe.
Okay, so thank you for the presentations and the questions afterwards. We move to our next speaker. So I'm now happy to announce a presentation by Vanessa Almendro Navarro and Sadiq Kasim from Danaher. I hope you have access, and we're looking forward to listen to your presentation.
Thank you. Yes, we are going to be talking today about risk-appropriate manufacturing for personalized gene editing technologies, following on the story that has been already presented this morning about baby KJ by Kiran, Becca, and our collaborators. These are our disclosures. And I would like to start our presentation by reflecting on 3 examples of experimental medicines that have changed the course of science. In 2012, Emily Whitehead was the first kid to be cured of relapsed ALL with an experimental new therapy called CAR-T. This treatment that led to full remission on Emily became the first CAR-T cell therapy that was approved, known today as Kymriah. And today there are around 10,000 patients that are eligible in the US to be treated with Kymriah. In 2019, Victoria Gray became the first person to be cured from sickle cell disease with an experimental therapy based on ex vivo CRISPR gene editing, a therapy that became Casgevy and that is today available to treat around 16,000 patients per year in the US. And just a few months ago, as we saw this morning, KJ Muldoon became the first baby to be cured from a severe form of urea cycle disorder with a personalized mRNA-based editing technology. All these treatments, Kymriah, Casgevy, and Cave, have proven to be effective in providing virtual, long-lasting cures to very devastating diseases. But in contrast to Kymriah and Casgevy, Cave could only be used to treat baby KJ with no traditional commercial opportunity. And while we have proven today the technical feasibility of developing these personalized gene editing technologies, not the traditional biotech or biopharma would have ever taken in manufacturing a treatment for a personalized patient. We need to change the paradigm in the way that we develop and deliver these personalized treatments to the patients. Delivering these scientific breakthroughs into medical advancements and treatments is going to require, as we all know, to change the way that we think on the development of these therapeutics. Importantly, we need to be able to develop these treatments for the right patient at the right time, at the right quality, and at the right volume. And that is going to require for us to think differently in the way that we develop these medicines. And importantly, we need to become much better in the way that we manufacture them. Cell and gene therapies are relatively new modalities that really require a fundamental shift in the way that we think about manufacturing these components. For example, unlike small molecules, genomic medicines are much more complex. They have multiple components, such, for example, a guide RNA, an enzyme, and a lipid nanoparticle. And they require optimization of every single individual component when it comes into manufacturing, and as well of the final drug product. This complexity in the raw materials and individual components of gene editing, cell and gene therapies in general, introduce as well different considerations from a supply chain perspective regarding raw materials, the logistics of those materials, and as well different considerations from the chain of custody. And importantly as well, manufacturing of these treatments requires a different way of thinking in the regulatory framework. We need regulatory frameworks that are dynamic and that can evolve in a step manner together with the dynamics in the science itself. And this is precisely one of the challenges that we have been taking at Danaher, considering the systematic challenge that we are trying to solve for the manufacturing of cell and gene therapies technologies. And with companies like Cytiva that has bioreactors, for example, with Aldevron, where we manufacture GMP mRNA, and with IDT, where we manufacture GMP for guide RNA, we have been taken into trying to solve these complex challenges of manufacturing gene editing technologies and many other personalized treatments. And the reason why we have taken into investing in solving for manufacturing challenges is because while the promise of genomic medicines, as I mentioned before, has been proven, we still are at the infancy of making sure that these treatments can deliver on the promise. We know that today still there are many challenges in identifying and qualifying the right patients that could be treated with, for example, personalized gene editing technologies. There is still significant unknown in the function of these mutations that could lead to selected targets to deliver clinical efficacy. From the manufacturing perspective, as I mentioned before, there is still the need to do significant innovation to increase the success rate of the treatments that we develop and then deliver to the patients. And we are, as I mentioned before, trying to take a systemic approach to solving all these challenges, but we recognize it has been expressed in the prior presentations that solving these complex challenges require collaboration. Danaher is not going to do it alone. There is not a single hospital or institution that can solve for these challenges alone. And this is precisely what we have been doing for the past few years, establishing strategic collaborations with additional leaders in the space and leading institutions so that we can solve for this end-to-end workflow challenge that we are facing on the gene editing space. One of the collaborations that we launched, as Fyodor mentioned, is a collaboration with the IGI Institute, where we are trying to develop a new playbook for gene editing technologies for ADA and HLH indications. And it's due to this collaboration with IGI that we got involved as well in collaborating with CHOP, Kiran, and Becca in helping to develop the treatment for baby KJ. I know that this, the background on urea cycle disorders has been presented this morning, so for the sake of the time, I will skip these slides. But as we mentioned before, this is a highly severe disease that presents in babies, where today the current treatments are only symptomatic, and where we know and we've seen and we have demonstrated today that gene editing technologies could pose a final solution for the treatment of these urea cycle disorders. I will skip as well this slide, but we know that the approach that we took for baby KJ was actually a base editor, trying to change, one single nucleotide in the sequence of the patient. And our contribution, and my colleague Sadiq Kasim will get into the details, was to help in manufacturing the components needed for this personalized treatment. What the challenge was that as we were entering into this collaboration, doing for the first time manufacturing for the first time a personalized gene editing treatment for a urea cycle disorder, there were many manufacturing challenges that had to be solved in a very compressed timeline. We had to do a new guide RNA sequence for the first time, synthesize a novel mRNA editor. We had to, together with Acuitas, design a novel LNP formulation and do a comprehensive off-target analysis. Of course, there has to be all the right alignment with the partners, including the regulatory agency that was crucial to advance this treatment in a timely manner. And critically, from a manufacturing perspective, the standard timeline to develop these treatments, it's around 16 to 18 months. We had to really compress the timeline to do this personalized treatment into 5 months. And that required Danaher to really advance and innovate in a different manner. And with that, I'm going to hand it over right now to Sadiq Kasim, the Chief Technology Officer for Technology Medicines, for gene medicines at Danaher, who is going to take us through the different innovations that we drive at Danaher to support baby KJ.
Great. Thank you, Vanessa. So, as Vanessa mentioned, the work presented by Dr. Musunuru and Dr. Rebecca Arntz-Nicholas was really unprecedented in terms of the speed and the collaboration that it brought to the table. This is just a summary of the timeline and the number of players that it took to bring this therapy. And I think it's that integration and collaboration across such a short period of time that enabled that risk-benefit based manufacturing approach. So in the next couple of slides, I'll show you some of the insights that we've gleaned in terms of the purity of the materials and the requirements. So this is just a reminder of what it takes from a off-target perspective. Functional genotoxicity packages required for clearing the IND. Kiran presented really effectively here, so I'm not going to double down on this slide. But one thing that I do want to highlight your attention to is the off-target confirmation pipeline, RAMP-seq. RAMP-seq is a multiplex targeted deep sequencing methodology that employs RNase-H-dependent PCR for library preparation to confirm off-target editing at previously nominated sites, and the sensitivity is can reach up to 0.01 editing frequencies. It was developed by IDT, but it's really served as a confirmation tool that enables definitive validation as an orthogonal method of off-target sites identified through other screening approaches, such as the one that Kieran mentioned. Built on top of that, one of the other pipelines that was developed in this context and that's now broadly available is UncoverSeq. So UncoverSeq, which is also listed here, it's an in cellulo method that shares the same core RHPCR technology, but it's specifically designed as an end-to-end in cellulo nomination system using NHEJ-based double-stranded DNA tag integration for initial off-target identification. So I think the key point here is that both methods use the same overall chemistry to achieve comparable detection efficiencies, but UncoverSeq functions as like the comprehensive screening tool that identifies candidate sites, while as RAMP-seq serves as the confirmation method. So I think what this slide highlights is that, the need for platforming in terms of developing a genotoxicity package that can enable proceeding into the clinic. And these are some of the tools. And I think between RAMP-seq, UncoverSeq, OneSeq, and ChangeSeq, I think this could be a really interesting battery of platform methods that can accelerate the path towards genotoxicity risk assessment. But in addition to off-target nomination, RAMP-seq has also been used internally for determining functional purity and functional efficiency of guide RNAs that are manufactured. So if you go to the next slide, so this just summarizes nomination and confirmation. We can skip this slide. We just talked about that with regard to UncoverSeq and RAMP-seq. This again is a summary. So from manufacturing perspective, how did we manufacture under a compressed timeline, something that typically takes 18 months? In this case, took less than 6 months. If you go to the previous slide, Vanessa, just wanted to talk through that before this slide. So one way that we were able to achieve that is because, as you know, there are three components to the final mRNA drug product. There's a lipid nanoparticle formulation, there's a guide RNA, and then there's the mRNA. And typically the way that the ecosystem is set up is that you have to go to a separate vendor for each of these components. So separate quality systems, separate audit trails, separate chain of custody, separate chain of identity. What we were able to do in the case of baby KJ and what enabled the speed is that everything was under one roof. And so that enabled single point of contact in terms of regulatory interaction and in terms of coordinating and harmonizing between quality systems. So with collaboration with Acuitas, the final drug product was manufactured in GMP compliant clean rooms under phase appropriate conditions. It allowed real-time batch review, enabled faster resolution and closing of kappas. And I think that joint expedition between the critical raw material, the drug substance, and the drug product really harmonized and streamlined quite a bit. So I think one point here is that having everything under one roof, which we were able to bring together here between Acuitas, IDT, and Aldevron, was really enabled operational speed. But in addition to that joint co-location, the next slide, if you go to the next slide, I think there was also constant partnership with the regulators across every stage of the process. So obviously the interaction was led by Dr. Rebecca Arns-Nicholas and Dr. Kieran Musunuru with the FDA, but they had brought us in as partners as part of that regulatory collaboration. And so the regulators at every step of the way had access to the way that things were manufactured, real-time updates in terms of methodology. And I think what this enabled is the creation of a CMC template to streamline documentation while protecting trade secrets, and the joint quality systems between the QA/RA teams delivered a 300-page technical package in 4 days, enabling rapid FDA review and direct communication with FDA to enable real-time resolution. So the point here is that it requires really hand-in-glove collaboration with both the clinical developer and the regulator in order to streamline things. So the next couple of slides, I'm going to zoom out now and talk about the critical quality attributes of some of the raw materials and their potential impact on overall on-target and off-target editing. So if you go to the next slide, Vanessa, one of the outstanding questions in the field right now is the role of purity in determining on-target gene editing efficiency. So what we did in the study here at IDT is we synthesized the same guide RNA, but using 3 different methods. So we looked at 3 different production methods using the same guide RNA sequence and 3 different manufacturing qualities, the RUO meaning not in a clean room, GMP meaning with clean room documentation, the manufacturing technology, the Cytiva ACTA processor versus synthesizer versus the IDT home created synthesizer, and then the time to manufacture for each of these technologies and the downstream purification method. So in 2 instances, it was HPLC purified guide RNA that was used in the on-target assessment here of the data shown below, and in one was a standard DSALT. And we quantified the purity using orthogonal methods, both mass spec and RAMP-seq. And here the specificity of the methods is highlighted. So I think the main takeaway here is that when you look at the overlap between the RUO HPLC and the GMP engineering run methods, here as shown as cell-based activity and in vitro activity in K562 cells. What you see is, once you reach a certain concentration threshold, the RUO HPLC guide RNA performs nearly identically to the GMP standard method, which takes 6 to 18 months, with all of the downstream purification, documentation, et cetera. Whereas there's a clearly market difference when you just take a standard DSALT with the 40% purity guide. So then when we looked at the relationship between the guide RNA purity and activity, here what we've done is we took the standard guide, which is 100 nucleotides, and then we mixed it with a ratio of n-1 guide, where there's a indel, a clearly introduced indel, and then we put this ratio together, and this generated a really nice linear relationship in terms of purity between observed and actual. So then we looked at the functional performance of these mixed ratio, the full sequence with the truncated guide RNA, and looked at the impact on overall on-target gene editing. And what you could see here on the right-hand plot is when you look at percent indels or percent gene editing versus the observed purity of the guide, that obviously at lower purity levels, you get less on-target editing. But there is a threshold where the on-target editing does seem to plateau, which is at about, here in this case, 70 to 80%. So here, I think this is a study illustrating that, sometimes, it's obviously the desires to get more pure material. But when you look at, if you go to the next slide, but when you have to make a trade-off between risk and benefit, I think the opportunity here is to define a framework that more comprehensively takes into account risk-benefit appropriate products, where if you're treating an N of 1 or an N of few indication versus a large indication like familial hypercholesterolemia, you accommodate release specifications that take into account both the critical quality attributes, the indication, and the scale. And this is different from the current CGMPs that obviously have more extreme requirements to balance safety. But here, what we're arguing for is to create manufacturing solutions that solve for scale, cost, and timelines, and to establish data for risk-based determination of the minimum viable product critical quality attributes for guide RNAs using preclinical comparability and clinical outcomes data, because this is a big gap in the field in terms of critical quality attributes that actually are meaningful from a safety and efficacy perspective. And I think introducing this risk-benefit sort of lens will help us to both treat more patients and generate more data, and then leverage that data across multiple trials. So the final slide just highlights some of those points to summarize from this presentation. Platformization is essential, especially for things like, the genotax off-target package, but then also the way that you detect off-target edits and the purity of the ancillary material and the critical raw materials. Risk-appropriate GMP, we believe, will unlock greater feasibility and a greater number of patients being potentially dosed. Having more patient-centered CMC analytics where we incorporate the patient's own genome into off-target analyses could represent a new paradigm of individualized quality assessment. And I think that using that data in a centralized repository of data will be needed for scalability, so that it's not just one-off academic centers doing their own thing, but it's collectivizing and federating that data in a way that's actionable by other developers. And I think the main takeaway here overall is that no single entity can deliver this alone, and the ecosystem models are really the future, and that ecosystem requires hand-in-glove collaboration from industry as well. So it's not just a government-academia partnership, but it's really a three-legged stool between these partners to really accelerate. And I'll close here. We'll open it up for questions.
Okay, many thanks for the presentation. Open for comments, questions.
Thank you very much for the presentation. I think one of the challenges that I see in what we've heard so far is, on one hand, we talk about treating single patients and the need to have more flexible requirements for those. But at the same time, we're talking about platformization, which would then result in multiple patients being treated. And at that point, the question is, how is it then still justifiable to have more flexible and reduced requirements? And for me, it's also the difference between treating a single patient and actually authorizing a medicinal product for wider use.
Yeah, Fyodor's been our comrade here, our partner. He's thought about a lot about this. Fyodor, do you want to take this?
Oh my goodness. Well, first, I'm glad Kieran and Rebecca are also on the line, and you, Sadek, and you, Vanessa. We've been thinking about this a lot. That doesn't mean that we have a good answer, but let me offer you the answer we have so far, and I'm very eager for your perspective. At the end of the day, the key critical quality attribute of any on-demand made CRISPR is how well it did clinically. That, by definition, is not a CQA you can measure in any non-clinical study. So we have, if you will, a walk before you run mindset. Here's what I mean by that. Consider an integrated manufacturing, non-clinical potency and safety assessment pipeline under which an on-demand made editor is assessed for purity analytically, and then you look at off-targets, nominate and verify, you look at potency using a cell-based model, and then benefit-risk considerations justify its administration to the first subject. And the subject does clinically well. Now you make a second one using everything exactly the same. So it's the same rig at Danaher, if you're working with Danaher. It's the same cell line model that you used for the first one, like everything is the same. We have argued that if the second product meets or exceeds the specs exhibited by the first product in these non-clinical and analytical assessments, then probably we should, get the go-ahead from the agency to dose the second subject. If the second subject does well, which is an if, great. I'm sure you see where I'm going with this with respect to subject number 3. If the second subject doesn't get well, unfortunately, okay, now we've learned something. I'm really glad you also underscored sort of the difference between clinical trial development and authorization. a lot is going on the latter front, and I don't want to take time this morning or this afternoon to speak to this, but we think that ex vivo non-clinical comparability assessments of product 1 to product 2 could potentially open the door in these accelerated manufacturing and de-risking frameworks to dosing multiple subjects sequentially, not parallel, of course, such that the agency gets more data, frankly, and gets either more confidence or gains questions with respect to whether or not this is the platform approach to follow. I'm super curious for what Kieran and Rebecca have to say. Sadek, you and Vanessa, I didn't mean to barge in here. I'm really glad you asked this because this is something we think about a lot.
Yeah, absolutely. Yeah, I mean, Kieran, yeah.
I guess that's my cue to say something. So I view it a little bit differently. So, you're talking about patient 1, patient 2, patient 3. I don't see them as different products. I see them as a single product. I see the parallel with something like CAR T, where it is the process that is really the unifying theme. And by definition, every CAR T therapy is a different product because you're using different cells. But the process is similar enough, and the efficacy and safety issues are similar enough that when you actually evaluate that product/process, you're doing it within the context of a clinical trial. And so I don't see this as a collection of N of ones. I don't think that's the way we should be pursuing this. We should do the work up front, get clearance via, an IND, if we're talking about the FDA or CTA, get clearance to do an umbrella clinical trial with essentially slightly different versions of the same drug product, but ultimately it is the same drug product. And in the fullness of time, it should be evaluated as any drug product is. Is it safe? And is it having desired effect? And I don't think anyone expects that every single patient who's going to be treated with any drug product is going to have a complete response. That's not true of pretty much any drug on the market, right? You look at the aggregate data and you assess, is there more benefit than risk with this product, just the way you would with any other drug product? And then at the end of the day, you might have to be a little creative with what the label looks like, but the marketing authorization should be around that product. So again, it's not a collection of different drugs tested in an N of 1 scenario. It is a single drug entity that is tested in a formal clinical trial design in a unified way where you're collecting data systematically across all of the patients who are enrolled in the clinical trial, and then you can make an informed judgment as to the safety and efficacy of that product and proceed from there. That's my view of things.
Thank you very much.
Yeah. I'm sorry, just one sentence. I need to blame sleep deprivation for having completely messed up what I was trying to say. I agree with Kieran. All I was suggesting is when you make product on a platform for a subject number 75, how do you know whether to administer it to a patient? You have to run some analytics on it. And I'm hopeful it's non-controversial to say, well, you have to compare some of its non-clinical features to what was used previously. I completely agree with Kieran that this should not be regulated as a constellation of N of ones. This should be a platform.
I think this is what we would agree on, and I think just again for the audience, I think there is also a difference between the US legislation and the European legislation in that single treatment, so emergency INDs, are still within the legal framework, whereas the exemptions in Europe actually remove the requirements from the medicine's legal framework. And I think that is one of our considerations, as I've said before, that if you focus on treating single patients in the exemptions, that doesn't generate the same quality of data as you would in a formal clinical trial. So I think the way to go forward is indeed the platform concept, where you consider that the manufacturing process is the uniting feature, although the product looks different for each patient.
I think with this statement, I want to call for a short bio break. Apologies for the delay, everyone. So we're ready to start.
All right. Well, I want to first thank EMA and the CAT for convening this important meeting. I'm Michael Lemke, the Senior Vice President of Scientific Affairs at the Alliance for Regenerative Medicine. I'm going to speak briefly today about leveraging data via platform principles in gene editing. You've heard about this already today, and you'll hear more details in the subsequent presentations by developers, which will follow mine. We really, at ARM, with our members and regulators and academic KOLs, explored these principles in a meeting that we call the Scientific Exchange in November of last year. We convened this meeting with the support of ISCT and Danaher to share best practices for employing platform principles in the development of gene editing therapeutics for rare disease. As you know, and as you've heard today, gene editing is uniquely able to address many minor genetic disorders, including rare diseases with multiple genetic variants. However, regulations do not formally identify how platform principles may be used to streamline development. So we invited academic KOLs, including some of the presenters here today, and some of the developers here today, you can see on the slide, to present case studies, both real and hypothetical, about how platform principles may be applied to the development of gene editing therapies. FDA leadership and experts were present at this meeting to listen and comment on the merits of the case studies, and EMA and MHRA were also present in listen-only mode virtually via a live stream. The results and the summary of this meeting have now been published in Cytotherapy recently, and we can make that article available to those of you who are here who haven't seen it. And we also had a brief commentary about the subject in Nature Biotechnology. And I'll highlight here just a few of the key themes that emerged. First and foremost, as you've heard a little bit about today, the consensus is that the delivery vehicle, whether it's an LNP or a viral vector or another vector, is the primary driver of biodistribution. And really this means if you can hold the vector constant, you should be able to streamline biodistribution and tox testing. Another theme is around manufacturing and fixing certain elements of the platform, fixing manufacturing steps in particular. And if you have common manufacturing steps, you should be able to streamline validation and release testing, realizing significant savings. On the clinical side, these applications, we think in particular, lend themselves to an umbrella trial approach. And this is really the most efficient and effective way to study diseases where there are multiple genetic variants. And last but not least, GMP requirements for guide RNAs, for example, should be applied in a risk-appropriate manner. And I think you heard a pretty good example of that in the last presentation given by Sadek. And the consensus was that if you could realize these principles on a consistent basis, these could translate into material efficiency gains on the order of 2 to 5 times. And as you've heard today, lead to development times in as little as 6 months. Some of the regulatory solutions or guidance that I think we need are defined criteria for what a gene editing platform is. Also, the ability to cross-reference prior CTAs or INDs with respect to CMC, preclinical and clinical data across related therapies. But going beyond that and developing evidentiary guidelines for expanding platforms across new indications or patient populations as we understand more about the platform and characterize it more thoroughly. And last but not least, allowing master protocol approaches with shared clinical infrastructure and long-term follow-up. So with that, I will stop and pause and see if there are any questions. And if not, I know we're a little bit behind. We can move on to the next presentation.
Okay. Are there any direct questions to Mike? If not, Mike, I propose we continue with the first presentation from Christy Wood from Intellia.
So Christy's going to speak about challenges in the development of in vivo gene editing therapeutics. And she comes to us from Intellia.
Good afternoon or good morning, depending on where you are. I am Christy Wood. I'm the Senior Vice President of Technical Operations at Intellia Therapeutics. And this morning, I'd like to speak about some challenges or maybe opportunities for development of in vivo gene editing therapeutics. Gene editing technology really represents a true paradigm shift in how we approach therapeutic development. Unlike traditional therapies that manage symptoms, what we're seeking to do is really to treat patients at the root cause of their disease. And this, certainly creates a lot of excitement around the potential to have single dose treatments with potential lifelong benefit for patients. And what we're, overall looking to do is to develop differentiated medicines using gene editing technology that create new treatment options for patients. With that said, gene editing can encompass a wide variety of permutations. So you can think about therapeutic approaches that we've been hearing about being either in vivo or ex vivo, autologous, or even allogeneic cell therapies. And with that, you can imagine that there's a variety of different editing tools, formats, delivery systems, and manufacturing technologies that can be used for gene editing technology. So you can imagine any kind of permutation that might be selected from this type of list that developers may be looking to use moving forward. And with that comes differences in requirements for both non-clinical safety and efficacy studies. And I think it's important to mention as well is that editing tool classification can actually range quite a bit from maybe being a starting material to a drug substance or even potentially a drug product. And that really changes the quality requirements around these therapeutics. So one of the big challenges, I think, is writing and developing a single regulatory guidance that covers all of these potential options for gene editing therapeutics. And so one of the things that we really want to focus on is risk-based guidance that really is derived from the science behind the technology that allows for flexibility as the technology continues to mature. With that said, Intellia has been able to progress multiple programs in vivo gene editing. We currently have 3 ongoing phase 3 studies. One is for the treatment of hereditary angioedema with NTLA-2002 or Lonvo-Z. And then the other disease area that we have in focus for us is transthyretin amyloidosis. We have 2 additional phase 3 studies, one in cardiomyopathy, which is a much larger phase 3 study, and then the final study in polyneuropathy, which is a little bit smaller. And this uses NTLA-2001 or Nexi. I probably don't need to describe this in depth, but we use a CRISPR-Cas9 cleavase system for our lead programs. I like to think of this as a software and hardware combination where that software is really the guide RNA and that identifies the genetic target. We've heard from other speakers, that's what can be swapped in these systems and really allows for the reprogrammable element of the CRISPR-Cas9 system. You can think of that Cas9 protein as the hardware, and in combination with that guide RNA, that's responsible for the targeted DNA editing and the enzymatic activity of this system. It's important to mention that CRISPR-Cas9 is selective. It can be developed to be highly potent, potentially addresses any site, and can also target multiple DNA sites. So the talk and really the focus of today is on in vivo therapeutics. So when you think about this, CRISPR is actually the therapy. We're delivering the CRISPR machinery in vivo, and we're doing gene editing within the body. Our focus right now is on genetic diseases, and there's several strategic advantages that we view from this type of technology and approach. Certainly the potential for single dose treatments. We also focus on systemic non-viral delivery of the CRISPR-Cas9 machinery. We think that offers several potential safety advantages in that you have transient expression of that editing system. We're focused in our lead programs on gene knockout, but you certainly have the potential to do gain of function by selecting or potentially changing that editing tool that you use. And right now we're focused on delivery to the liver for these lead programs, but many developers are working on delivery systems that can expand that to tissues outside of the liver or extrahepatic applications. To dig in just a little bit more, I want to focus on the in vivo knockout CRISPR mechanism of action for a liver LNP-directed therapy. So we have a lipid nanoparticle drug product that encapsulates a targeted gene-specific guide RNA along with a Cas9 mRNA. This is delivered by an IV infusion to the patient. It's taken up in hepatocytes through an ApoE-dependent or LDLR receptor-mediated endocytosis mechanism. You have endosomal escape and release of that CRISPR machinery in the cytoplasm of cells where that Cas9 mRNA is translated to Cas9 protein, associating with a single guide RNA, forming that RNP complex, which translocates the nucleus. It's then able to find the complementary genomic DNA target site, at which point we create a double-stranded break. And through endogenous DNA repair mechanisms, it leads to an indel formation, essentially knockout of that gene, with the final result being a reduction in the level of circulating plasma protein. So the direct action here is the genomic edit, followed by the intended effect in our case, which is reduction of a circulating plasma protein. Okay, so now that we've covered some of the basics, I guess, around gene editing, I want to go through a few different case studies and what we've discovered in our development pathways. And the first is focused on some clinical considerations. So right now we have global CTA submissions that cover 3 different phase 3 trials, as I mentioned. We're in greater than 30 countries around the world with our clinical trials. We've had more than 15 regulatory authority meetings to date. And in those interactions, we've received greater than 300 queries, and I would say that number continues to increase quite a bit. And one of the things that we've realized is that each country really has unique requirements. As we've approached a global regulatory submission, it needs to be well coordinated and strategic as you think about how to kind of harmonize this across many different countries. So, from a country-specific perspective, each regulatory authority certainly has unique requirements for documentation, review, and approvals. We've seen differences in standards for safety, efficacy, and quality that may vary from country to country. What this leads to is a lot of requests for additional information during CTA review. We've also seen differences in post-approval safety reporting. One of the things that we've learned is that local advisors certainly play a crucial role in the communication between sponsor and regulatory authorities. In terms of harmonization, ICH efforts have certainly harmonized many regulations, but we still see many differences, as I was alluding to. One example has been around GMO regulations. And then certainly this isn't necessarily unique to gene editing, but submission documents need to be adapted to local language as well as, provided with some cultural context. So there's a lot of translation requirements. And I think, where that's impactful in the space of gene editing, where you've got a lot of novel technology, that can add a lot of time and a lot of complexity in communication between sponsor and the regulators. And just overall, this requires a lot of meticulous project management in order to manage multiple simultaneous submissions and also to keep these submissions harmonized across all of these different countries. And so the best laid plans certainly aren't always successful. And this is just a few examples of where we've seen some challenges. But we've had primary follow-up and secondary follow-up clinical protocols reviewed by different divisions within the same health authority, which resulted in conflicting guidance. So opportunities for improvement there. We've definitely seen prolonged review timelines due to several rounds of informal and formal review of responses. Some of this, can be attributed to a lack of direct communication between sponsor and reviewer, but certainly opportunities for improvement there. And then, in another specific example, we've had approval of the clinical protocol that we've been pursuing in over 25 countries, including the US and EU. We had one particular health authority provide a significant number of unique changes that were requested in the clinical protocol. And I think that makes it very difficult for a sponsor to, again, run a single global clinical trial when you have so many different unique requests. So we see some opportunity that may exist for mutual recognition for global protocols or even enhancement and regulatory flexibilities that really allow us to move these therapeutics safely and provide different country populations access to these potentially life-changing therapeutics. I'm going to go through 2 additional case studies here. We've had quite a bit of introduction on guide RNA purity and impurities and off-target approach, and I'll speak a little bit about that. And I'll also touch briefly upon an impurity control strategy for a novel excipient. So just as a reminder, for an in vivo knockout product, this requires 2 drug substances, so like guide RNA, which identifies that genetic target, as well as a Cas9 mRNA drug substance. We have an LNP drug product that contains both of those RNA drug substances. And within our LNP drug product, we also have a novel lipid excipient. At Intellia, we really leverage synthetic manufacturing across both drug substances as well as drug product. So that guide RNA is typically 100 nucleotides in length for our lead programs. And the specificity of that to the genetic target is defined by the 20 nucleotides at the 5' end of that guide RNA. And this uses standard solid phase oligonucleotide manufacturing processes. In contrast, we have a second drug substance in Cas9 mRNA. We use an enzymatic synthesis approach. It's approximately 4,400 nucleotides in length, and that encodes for the SpyCas9 protein. In addition, we have multiple lipids that are used within our formulation, including compendial, non-compendial, and that novel lipid excipient that I mentioned previously. So a variety of different regulatory requirements around those excipients. And as far as the LNP drug product goes, we combine both of our RNA drug substances in that LNP, and this is made by a self-assembly process with controlled dilution. As you look at each of these manufacturing processes or platforms, each of them requires about 15 to 20 analytical methods on their own, really for release of those components. This also includes multiple cell-based assays for both drug substance and drug product. So I think you can understand, just from this system itself, there's a lot of effort that sponsors are putting into developing both the manufacturing and analytical methods with which we use to manufacture and control these products. So I'll go into a little bit more depth on guide RNA manufacturing. This is just a generic process overview of an upstream and downstream oligonucleotide manufacturing process. These are typically made in the 3' to 5' direction. And in terms of previously marketed products such as siRNA or antisense oligonucleotides, we're actually using the same type of oligonucleotide manufacturing that is used in many of these commercial products. And what that means is that we have standard phosphoramidite chemistry with similar raw materials. And the approach to the overall process and control strategy are really independent of oligonucleotide length. What we've realized, though, is that there are many key challenges in development of guide RNAs from both a process and analytical perspective that do require optimization for long-mer oligonucleotides. We are of the belief set, though, that an established guide RNA process can be applied across target sequence changes. And just to highlight a little bit more around the technical challenge of manufacturing of guide RNAs, if you understand this process, it is a nucleotide by nucleotide addition where the purity is related to the cycle efficiency of that nucleotide addition from a theoretical perspective. And so if you just compare, say, a traditional 20-mer with a 99% cycle efficiency, you see that crude product purity would be 82%. In contrast, when you're developing 100-mer, again, if you assume that same cycle efficiency of 99%, you would be left with a crude product of 37% purity. So we rely heavily on purification to improve the overall purity of these products. And you can also understand that there's an increase in the overall possible distinct impurities that can be formed through this manufacturing process. So there's definite challenge and as well as a lot of opportunity for synthesis optimization, as well as improvement in purification technologies and recovery or overall yield. And we've also had to develop a number of analytical tools along the way, a new methodology that allows us to better characterize the impurity profile, as many of the methods developed for 20-mers or 21-mers may not have been fully applicable. One thing that we've gotten feedback on is that full-length product purity should be greater than 80%. This is a recommendation that had come from the FDA. And so this has been a focus for us, certainly along our development trajectory and really improving overall purity. But I would say also control of that impurity profile as we've moved from early stage into late stage clinical development. And it's worth highlighting that, product purity is inversely proportional to product yield. And so that's something that, needs to be taken into consideration in terms of development. So as I was mentioning, improving product purity is something that we sought to do throughout the development lifecycle. I'm just highlighting here that as you implement process changes, this does require a formal assessment of comparability. And our comparability assessment included release data, extended characterization testing focused on guide RNA activity, as well as a detailed impurity analysis, as well as stability comparisons, looking at process 1 versus process 2, just showing the chromatogram on the left, again, improvement overall in full-length product purity, as shown in process 2 in red, and a reduction in the total impurities. On the right is just an example of a thermal stress, side-by-side look at these impurity profiles after that thermal stress. And what you see is a consistent impurity profile from both process 1 and process 2 that helps us understand that there's similar degradation pathways in these products, and the overall chemistry is not changing between process 1 and process 2. I think naturally, as we've already heard about, this leads you to think about on-target editing as well as off-target or potential impact to the off-target editing profile. So outside of the formal comparability, we do look at both on and off-target editing. For us, we use primary human hepatocytes. Again, our tissue of interest is the liver. And from a verification perspective, I'm showing some data comparing both process 1 and process 2. This is across multiple PHH donors. We treat these with the formulated LNP drug product containing guide RNA made from process 1 and process 2. In the example in the top panel, we're looking at on-target editing for KLKB1, which was our intended on-target locus for this product. You see a dose-responsive increase in editing. You also see that there's no difference in the editing profile for either process 1 or process 2. And then I'll direct your attention to the bottom panel there. MAPK1 is a verified off-target at supratherapeutic doses for this target. And then again, looking at both process 1 and process 2, we show no difference in that off-target editing profile, helping to demonstrate consistency between these manufacturing processes. We think it's incredibly important to develop a risk-based framework to help sponsors determine when off-target editing should be conducted in primary cells. These are challenging assays to do and certainly very time-consuming. And so I think it's important that we focus on the ability to demonstrate batch-to-batch consistency through analytical controls. You also can use your data, as you've done on and off-target development, and understanding the impact of that guide RNA product purity or even grouped impurities that you can vary in these analyses. So, the example we're giving here is showing consistency even with process improvements where we've actually improved the purity of this product quite a bit, and you see no difference. Also, the ability to do supersaturation here well above the exposure in patients also helps create that safety margin and understanding of potential impact. Overall, an impurity classification system and development of that really helps define the potential impact to the off-target profile from individual impurities. And so that's another way to approach kind of a risk-based framework. In an example, a 3' truncation may not be active and would have no impact on that off-target editing profile, whereas you might treat a 5' substitution or a 5' truncation a little bit differently. And so that helps you define what that impurity control strategy might be for individual impurities that you're detecting in the manufacturing process. So I'll switch gears quickly and just go through one additional case study, but it's really around, again, impurity control strategy, but development of that for a novel excipient. We use an ionizable lipid excipient. This is a synthetically manufactured small molecule. We've used the same route of synthesis throughout development. It's a 2-step manufacturing process and one of 4 lipid components that we use in drug product manufacturing. It's classified as a novel excipient, and that really requires the same level of rigor as a drug substance. And so as we were developing our control strategy, specifically around the impurities, we looked at ICH-Q3A, to understand reporting, identification, and qualification thresholds for those impurities. And the table on the right is directly out of the guidance document. And I think what you'll see is that you see a maximum daily dose highlighted. And then within identification and qualification thresholds, you see milligram per day intake. And I think it's something we realized early on that there's really minimal consideration for a single dose therapeutic and how to apply ICH-Q3A. So it's something we sought health authority feedback on early in our development life cycle, And we found a lot of inconsistency across agencies in terms of how we might approach that impurity control strategy. And, really what we want to focus on and what we're pushing for is, patient-centric impurity control strategies, certainly in relation to ICH Q3A. And the table on the left is really derived from the appendix or the ICH Q3 decision tree present in the guidance document. But we think it's important that an impurity control strategy, certainly ensures acceptable product quality with an intended use and focus on safety and patient needs. And kind of in the spirit of taking into account ICH Q3A, do you understand any of the identified impurities from a structural perspective? If you don't, are there any ways to group those? And how can you, review that with existing process knowledge? Pairing that with duration of use and maximum exposure to the novel excipient is important as you develop that control strategy in really looking at your toxicological assessments, whether or not that be general toxicology studies or genotoxicity studies. And then certainly as you get further on in development, being able to leverage your clinical experience across programs is important in developing that impurity control strategy. But in general, for in vivo gene editing drug products, where they're designed essentially as a single one-time dose, you have a, overall reduction in that toxicological burden. So I think there's opportunities for improvement in, directing sponsors and how to use some of these guidance documents and maybe thinking a little bit differently, especially early on in development when you may not have as much known about either the process or the analytical methods. So just overall, there's a lot of significant time and resources that are used in initial platform development. It's important to be able to leverage that prior platform knowledge in current and also future programs. There are a lot of regulatory learnings and interactions that we have seen around the platform, and we want to be able to leverage that moving forward in development of therapeutics. So just a few key takeaways. We believe gene editing regulatory guidance needs flexibility just due to the technological diversity that sponsors are using. Global regulatory harmonization certainly remains a challenge for moving these therapeutics efficiently to patients. And we think there's, opportunities for single dose therapeutics, so we may want to rethink some of the safety assessments and approaches to impurity control strategies. Really with recommendations to focus on risk-based guidance that allows some flexibility as the technology continues to mature. Another opportunity may be consideration for specialized reviewers for ATMPs who are educated on the complexity of gene editing technologies. And one of the things that we've also, think could be an opportunity is establishment of mutual recognition across countries, especially as programs advance into phase 3 and you're developing global clinical trials, allowing for better harmonization within that clinical trial. And with that, I will take questions.
I have one. You referenced that the feedback you've had from FDA is that purity of the guide RNA should be greater than 80%. I believe historically that's based on the shorter oligos, the 20 MERS. And you kind of hinted at this, but you didn't say it. Do you think there's a case for lowering that threshold based on the other work that you described?
So, they have spoken at, town halls and have given some, I think, absolute numbers. What I would say is it's absolutely dependent on your analytical method. And I think we all know that the same analytical method cannot be used across every guide RNA. There's different chemistries that are in use, and those methods have to be tuned to the particular guide RNA. So I think more importantly, it's you need to understand that impurity profile, and the control of that impurity profile is really important to develop both a safe and efficacious therapeutic. So I'm less stuck on the overall purity, but certainly, as we've gone through development, we've actually been able to improve that manufacturing process significantly where we have improved purity and still maintained yield in the process that it becomes developable for us.
Any other questions? There's a hand raised.
There's one question from the floor.
Yes,
here.
so it's me. Thank you for the presentation. And I was wondering, you said that you see the CRISPR enzyme as the hardware, but maybe I didn't understand it correctly, but it depends also on the pump sequence which enzyme you can choose best. So is it really
Absolutely.
hardware?
It would be hardware if you're using the same enzyme. Certainly, if you're changing enzymes, I don't think that hardware analogy applies.
Any last questions? So if not, we'll move on to the next speaker. Luis Barrera from Beam Therapeutics will speak about base editing opportunities for platform development.
So good afternoon. I'm Luis Barrera. I'm here representing Beam Therapeutics, where I'm Senior Vice President and Head of the Research Platform. And thank you for the opportunity to present today. We'd like to share this case study where we're looking at base editing opportunities for platform development, especially as applied to inborn errors of metabolism and other monogenic diseases. Just as a very quick refresher, Beam's primary gene editing technology is called the base editor. And you've heard a lot about CRISPR and different modalities. This is one of the flavors that you can take a Cas9 or another CRISPR enzyme, use its DNA targeting ability, but modify its ability to make double strand breaks and instead keep the programmability of the guide RNA, which can tell it where to go in the genome. But in the case of a base editor, the role of the Cas9 is limited to exposing a short sequence of single-stranded DNA that can then be acted upon by the second component of the base editor, which is the deaminase. And this is an enzyme that will chemically modify this exposed single-stranded DNA and make permanent substitutions from one base pair to another. There's two main kinds of base editors. One is called the A2G base editor, or ABE for short. And in this case, there's a deaminase enzyme, usually TAD-A, that will convert an adenine to an inosine, which will ultimately be repaired as a G by a DNA polymerase. The other type of base editor is called the C2T base editor, or CBE. And these editors will use a different class of deaminases to perform a similar reaction, deaminating cytosine to convert it to uracil, which will ultimately be repaired as a T. So different types of base editors can achieve these substitutions and install them permanently. And with this, we're able to make all transition changes between bases in the gene. So at Beam, we believe that base editors have the potential to address many diseases that are caused by single nucleotide variants. And one example that is of particular interest to us is across different types of inborn errors of metabolism. And what makes these kinds of diseases appealing is that they're often, the cause of the disease is often centered in the liver, which, as you've heard, has now become increasingly amenable to delivery via lipid nanoparticle or LNP. And so I'll talk a little bit more about Beam 301 in the next slide, but it is an investigational therapy in clinical stages that is following this paradigm that you've heard about before, where you co-formulate an mRNA that encodes the base editor and a guide RNA along with several lipid components into an LNP. You can then infuse this in a patient where the LNPs will predominantly go to the liver. And then once the base editor is translated and finds its guide RNA, it will be able to perform its function and go and make single nucleotide changes at the intended site. So to give you a little more background on glycogen storage disease type 1A or GSD1A, which is the primary disease in which we'll be focused in this presentation, this is an inborn error of metabolism that affects an individual's ability to convert glycogen back into glucose, which is a normal part of glucose homeostasis. It is essential for being able to maintain relatively constant levels of blood glucose. And individuals with GSD1A have both of their copies of the G6Pase enzyme, which normally carries out this step in the pathway, and are therefore unable to regulate their own glucose levels. There's no treatment beyond dietary management. Essentially, these patients are often given cornstarch at specified intervals as an attempt to maintain glucose levels. But if their blood glucose drops, they can have seizures and even death if they don't find a dietary way of restoring their glucose. So Beam 301 focuses on a specific mutation that can cause glycogen storage disease type 1A. And this is a coding change that is changing an arginine in position 93 into a cysteine, also called RD3C. And this is a relatively prevalent pathogenic variant that is the cause of this disease. However, even though RAD3C is one of the most common variants, like many monogenic diseases, GSD1A has a diverse mutational spectrum. And if you look across ethnicities, even though RAD3C is the most common mutation in Caucasian individuals or in Ashkenazi Jewish individuals, as you look at different populations, you will see that there are different mutations that drive the GSD1A incidence in those populations. And in many cases, it might seem, for example, there's an RAD3H mutation that is relatively common in individuals of Chinese descent. And you might think, well, could we potentially use the same editor and guide? It's the same amino acid. Turns out that as much as it feels like you should be able to do it, the base that you need to modify in order to correct this mutation is actually on the opposite strand from the RAD3C mutation. So even if it's right there, the challenge is that for many of these mutations, if we really wanted to take the same LMP and go and address some of these other mutations, each of these would essentially be its own drug product. And this is the key challenge, right? Because it is a very different situation if we were to think about all of these mutations, some of which are quite rare, as completely separate development pathways. So this will be the theme for the presentation, which I know you've heard before, but I hope this will make it more tangible. And so the challenge starts at the very top of the funnel, and I'll just briefly describe how we think about lead discovery for correcting a disease-causing mutation. So in this figure, I'm showing you a sequence that is part of the gene that encodes the G6P ACE enzyme. And here in the middle, you can actually see the one base that is causing the disease-causing mutation, the RAD3C. And like I said, base editors, they function by exposing a short stretch of single-stranded DNA so that the deaminase can act chemically upon it. And so what we're typically limited to do is to tile the region and use different guide RNAs and different PAM variants. So to the earlier point, in this case, the base editor is acting partly like software in that the sequence is changing depending on which guide RNA we pair it with. And what we've ended up finding as we've done several programs targeting different disease-causing mutations is that we typically end up picking a different editor to really try to get maximum potency and maximum precision in terms of the base that is corrected. I'm going to very briefly switch gears to a different pathogenic mutation that proves the point I'm trying to make a little more clearly. So bear with me for a second. So this is looking at the sequence that harbors the mutation known as PIC, that is the cause of alpha-1 antitrypsin deficiency. And one challenge with this sequence, as you can see, is there's a bystander base right next to it, 2 bases away, that if we were to also modify that base, we would introduce an additional amino acid substitution. And so for this particular mutation, we engaged in a pretty extensive lead discovery campaign where we sought to vary several parameters that are tunable in base editors, like how many deaminases you have, the deaminase variant, the PAM variant, and ultimately generated a series of different base editor variants that achieved different kinds of edits that corrected the intended mutation precisely, or sometimes introduced this amino acid substitution that we believe to be functionally neutral. And so what I'm hoping to show with this slide is if you are limited to having to reuse exactly the same editor to treat different mutations that are the causes of the same disease, you might have to make some trade-offs in terms of potency or the exact editing outcomes you can get. So we're hoping to find some common ground with regulators and thinking about if an editor is minimally modified, can this potentially be amenable to a platform concept and we can reuse some of the same data and learnings. So one of the challenges for developing therapies for this kind of disease correction approach is that you typically want to have an in vivo model that fully models the disease. So ideally, you want to develop a mouse where the human sequence harboring the disease-causing mutation is present. It's a fairly humanized representative gene. And in addition, you have a phenotype that you can recover in the mice. So for the RAD3C and Beam 301 program that I mentioned, we actually did make a mouse model where these homozygous mice for the mutation, you can see that they have metabolic effects, in this case summarized by their smaller size. And we were able to show the PD effects of Beam 301 and show that these mice, if treated with Beam 301, you can actually rescue the phenotype that is driven by that disease-causing mutation. The challenge is that this is not often scalable if you're really looking to treat the full spectrum of rare mutations behind a disease. You can get some efficiencies, for example, if you think about making compound heterozygote models, but ultimately you run into challenges where as we get closer to any post-1 therapies, it becomes challenging to make a mouse model for every single one of these. And our belief is that this may not actually be necessary, that at some point, once you've established proof of concept in one in vivo model that is representative of the disease, you can look at additional mutations using in vitro assays and have a reasonable expectation that if you are to correct those other pathogenic mutations, you will see a similar phenotypic effect. When it comes to the toxicology package, I'll try to just summarize here. I try to list certain studies and their purpose and the species in which they're typically performed. This is for an in vivo base editing therapy using an LNP. And what I'm trying to show here at the big picture is that many of the studies that you would typically include as a toxicology package can really be thought about as LNP platform studies. So for instance, a lot of the genotoxicity assays, they're really looking primarily at the lipid components. And so if you are to reuse those components in an LNP that is loaded with a different base editor, mRNA and guide RNA, the reality is that many of these studies, including genotoxicity, cardiovascular safety, and the GLP tox study, should be able to inform each other. And of course, we recognize that in some cases, and especially for the first mutation in a given disease, it will probably remain important to have a disease model that allows you to assess PD, as well as some safety endpoints that are disease relevant. When it comes to pharmacology and biodistribution, you can do a similar grouping. So a lot of the biodistribution studies for LNP-delivered gene editing therapies, they're primarily looking at the distribution of the lipid and the mRNA, and this is something that can be done regardless of whether you're using the same guide RNA and mRNA in a different LNP formulation for a different mutation. Again, there are some product-specific studies that might be required. For example, if it is important to assess the biodistribution of on-target editing, it may be important to do that at least once or twice and confirm the consistency if, as we expect, the LNP is the primary driver of the biodistribution. Off-target analyses are an important component of the safety assessment for in vivo base editing therapies. And here I'm showing several categories that are typically done when it comes to the full off-target package. So one type that is unique to base editors is what we call the guide RNA independent off-target editing. And this is basically looking at the risk that a deaminase itself acting independently of the Cas9 and the guide RNA could go and deaminate bases elsewhere in the genome. We found that particularly with A base editors and also with new generations of C base editors, this effect is not observed, but we consider it an important part of the package. And this is something that as long as two products are using the same editor but a different guide RNA, or maybe even a highly similar editor where you're only changing its DNA binding preference, these studies should be usable across investigational therapies. For other kinds of off-target concerns, this is typically not something that you see with base editors in terms of genomic rearrangements because you're not making double-strand breaks, but we do them out of an abundance of caution. We also have guide RNA-dependent off-target editing. This is the typical case where your base editor could find sequences that are similar to the on-target sequence and make edits there. We do think this is ultimately a type of assessment that will need to be done for each guide RNA and editor pair because it doesn't really translate between different editors and guides. And there is also typically, if off-target sites are detected, some risk assessment that has to be done at the level of individual target sites or edits. CMC is another area where we can imagine many important efficiencies that would help drive this platform concept. Of course, in terms of the manufacturing process, we envision that multiple products could be developed using the same or highly similar manufacturing processes, and these would benefit from being able to share qualification or validation processes and results. Similar argument applies with analytical methods. We think there are opportunities for doing platform assay validations, especially for purity assays that should apply across LNPs. Although we also recognize that some product-specific assays might be necessary, especially when it comes to potency or identity. Instability is another area where we would expect different LNPs targeting different mutations, but using generally the same LNP should be able to inform each other. When it comes to clinical development, I won't go through everything here, but essentially we believe that gene editing therapies have the potential to give really large clinical effect sizes that might enable a reduction in patient numbers and may eliminate the need for a placebo arm. I suspect many of you have seen the Casgevy data from Vertex, for instance, and the effect that you see in terms of VOC reductions and other endpoints is quite dramatic. We think that this would be the case for many other gene editing approaches, and that also highlights the potential to use biomarkers as the basis for accelerated or full approvals, depending on the situation. Ultimately, one other aspect that could help clinical development is being able to leverage the safety database from the same LNP targeting different mutations across different patient populations with their own mutations. And as many of these are rare diseases, that kind of efficiency in terms of being able to leverage the safety database from across patients would be quite beneficial for clinical development. And then there's other efficiencies one can think about, for example, when it comes to the safety and dosing considerations as you go into the clinic. We think that many of these are going to be driven by the LNP. So when you're thinking about infusion-related reactions, potential increases in LFTs, immunogenicity, the information that you gain from the LNP developed to treat a given mutation can probably be leveraged effectively for other LNPs with the same components targeting different mutations. And so, for example, this could help inform first-in-human dose. It could help inform stagger. If these issues were not observed with a given LNP, this could help inform what those considerations would look like for others. Similarly, for mRNA and guide RNA, we think especially for the mRNA, there is limited risk for acute toxicity. So as long as we're seeing evidence of that with one LNP, we don't have a reason to expect that a similar LNP with a slightly different mRNA would show a different result. For guide-dependent off-target effects, we typically don't expect these to contribute to acute toxicity, so it's less of a consideration, but depending on the profile, there might be product-unique considerations that arise. And then lastly, I just want to point out some of the potential synergies that are achievable via master protocol and a potential umbrella trial. I think these should be fairly apparent that if you're treating the same disease, but it's caused by different mutations, you can have a lot of efficiencies in terms of investigators and sites, endpoints, inclusion-exclusion criteria, and so on. And so just to wrap up, I wanted to highlight some of the challenges that were discussed in this case study. So as I described, we think base editing therapies have significant potential to treat monogenic diseases. However, each mutation may potentially require the development of bespoke editing reagents, and having to treat each of these as a truly separate drug product would increase the development burden significantly. One of the aspects in which these manifest is having to develop in vivo model for every mutation, which may not be feasible. And then there's a lot of efficiencies that may also apply in terms of non-clinical studies, analytical methods, manufacturing processes, and a similar degree of efficiencies that are helpful in clinical development. So I will stop there. Happy to take any questions.
Thank you, Luis, for that excellent presentation. Any questions?
If I can make a brief comment, I think quite a few of the issues raised, I think, are solvable. But I want to emphasize another difference between US and Europe, and that is while in the US there is an expectation of positive benefit risk that's acceptable for an accelerated approval. In Europe, for conditional approval even, we require a demonstrated positive benefit risk. So I think that is a crucial difference, and especially as you were referring to biomarkers, potentially surrogate biomarkers, I think this is where you might see differences, and that is anchored in the legislation.
Thank you. That difference is worth pointing out. I appreciate it.
Emily?
Yes, thank you. A bit of follow-up comment to what Ilona has said, and obviously not only applicable to your talk. Thank you for your talk in any case, but applicable to, I think, all the things that we've heard so far today, is that with respect to clinical development and confirmation of efficacy and safety, I do see quite some challenges to robustly show that. Actually, I think if you have a disease where any halted progression, for example, or any improvement of the disease could be unequivocally attributed to the treatment because there is no spontaneous recovery whatsoever, that of course makes it a bit more easy. But we have a lot of conditions that are quite heterogeneous, where maybe the type of mutation determines the severity of the clinical phenotype. And I think then it would be very challenging to really show efficacy. And I was also triggered by Ilona's comment, because in the beginning, I think it was mentioned that the FDA would base their decision on a plausible mechanism or something like that. And I also foresee that would be problematic in the EU, and not even, I mean, that's not the scope of today, but it has crossed my mind on various occasions that from the perspective of reimbursement, that would of course be a whole different story. And I also see some challenges there. So just a nuance a bit to the platform approach from the clinical perspective. Thank you.
But if I can just briefly jump in, Emily, I think the discussion at the time on FDA approval based on mechanism was for the emergency IND. It was not a clinical trial. It was not a marketing approval. It was for the emergency IND. So that's slightly different.
Yeah, thank you for the feedback. It is an ongoing discussion. As previous speakers have mentioned, it is a new paradigm where we're addressing the root cause of the disease. And of course, reversibility is an important consideration. We'll have to understand how clinical benefit plays out in different situations. But we do think there's an opportunity there that once you've established the relationship between the correction and clinical outcomes for some mutations, you could expect others to have similar effects.
Theodore, you have a question or a comment?
I do. Thank you, Luis, for a spectacular presentation. Folks, I don't want to be blunt, but because I'm a professor in academia, I can say the following. I think the for-profit biotech sector is in trouble. not of their own making. Like Casgevy has been approved. That is great. I hope Verve gets approval. I hope Beam and Prime get approval for the 2 or 3 indications they're pursuing with specific mutations. I don't know what else is going to get approved for gene editing anywhere in the world, Western world, in the next 5 years. And so I want to really encourage the agency to separate 2 challenges. One challenge is how do you get something to full approval and licensure, which I think is a big topic that we should all be discussing. But I really want to underscore Luis's call, if I may, Luis, I hope I'm not misrepresenting what you said, that for clinical development, to get enough data in terms of what matters and what doesn't, this long tail of point mutations, I mean, nobody will ever work on them. I really want to be very clear. None of these subjects will ever be enrolled because under the current framing, swapping out the guide, I totally hear the fact that in Europe you can put multiple guides in, but going back to what Kieran and Rebecca were discussing with respect to different editors as well, this vision that we set out in collaboration with the ARM is in clinical development, not for approval. We need to get to a world where people should be treated with bespoke editors to determine what matters for safety and efficacy. How you then get these platform things, however you define platform, to approval is a bigger question. But in the near term, the biggest need I see is getting, let's say, Beam's technology to treat another 50 subjects, each with a bespoke editor, rather than, arginine 83 to cysteine. So I just really, forgive me for sounding like a broken record. I'm rehashing what I said this morning, but I think the Beam story is a great example of why innovative approaches and flexibility in clinical trial design with respect to who gets enrolled and what the CQAs are of the APIs is a really good example of where I think the field needs a lot of help from the regulators.
Again, if I
Thank you, Peter. Well said.
Sorry. Again, if I may jump in, I still do not see a principal issue, but the one wording I did also pick up is we have no reason to expect differences. I think that wording is problematic for regulators because we need to see some data to assure us that the claim of no expectation of differences is actually supported by data. So I think that's where the regulators have the most significant issue. If you have patient-specific products, where is the point where we can agree that there is a sufficient body of data that we are justified in our expectation that there are no differences? And I think this is where the science needs to support the regulators to help us make those decisions.
Yeah, thank you for that clarification. Do we have time for one more question, or do we need to move on at this point?
Is there a question?
There's a hand raised.
Very quickly.
So I had a question then.
Very quickly, please.
A follow-up question. So what would be the minimal set that would be then acceptable or would be proposed by applicants? I can assume that if you have once shown that changing the editor a bit does not matter so much, but I could imagine, for instance, that you do an immunology screening, that changing the editor a bit would not result to, for instance, an immunologic type of antigen. And of course, the off-target should be included. So what kind of... So I think that's what we need to think about. What do you need to show then in an overall package, or the first one, and then what can you what is the minimal package to be shown in the next product that is a little bit changed?
Thank you. I think in that case, what we would like to show is in vitro data supporting the idea that the important properties for an editor's safety or potency did not change. So you mentioned off targets. I do think it would be necessary to do a complete off-target assessment for a new editor, but we think it could largely be de-risked in vitro. And when it comes to immunogenicity, similarly, I do think it would be important to ensure no peptides are created that suddenly have a much higher probability of MHC binding, and some of that work could be done both in silico and in vitro. So I think it would be a matter of figuring out what that data package looks like, but my expectation is that it could be significantly driven by in silico and in vitro arguments.
All right. Thank you, Luis, for that great presentation and fielding those questions. We're going to move on now. We have a speaker from Prime. Apologies, but I don't have a name. Speaking on modular platform for in vivo LNP formulated prime editors targeting rare diseases of the liver.
Hi, and my name is Andrew Anzalone. I'm the scientific co-founder of Prime Medicine, and it's a pleasure to be here today at the workshop and present on behalf of Prime Medicine, and thanks to the organizers for inviting us. I think you're going to share slides from your end.
We can certainly do. So just say next slide, and I will move the slides.
Yeah, great. Thank you very much. You can go to the next slide. Today I'll be talking about prime editing in the context of liver-targeted lipid nanoparticles for rare genetic diseases of the liver, but I think a lot of what I say hopefully will apply to other delivery platforms and also other gene editing technologies. And some of the themes you'll hear from me today are, I think, consistent with what Luis just presented and others have iterated here today. And I thank you all for your comments and discussion so far. So to provide a little context for what I'll share today, I just wanted to give a brief overview of prime editing and some of its key attributes, and also a little bit about how it differs from other gene editing technologies. So prime editing is a technology that can target a particular location in the genome where a mutation exists, and correct that mutation back to the wild type or healthy DNA sequence, and thereby sort of correct or restore the function of that gene, and also maintain the endogenous regulatory control of that gene's expression. So prime editing uses two key components as illustrated on the left. The first is a prime editor protein, and this protein is composed of two domains. One is a Cas-Nickase domain. This is the DNA binding domain of the editor, and the other is a reverse transcriptase enzyme. Importantly, the Cas-Nickase domain has been engineered so that it only cuts one of the two strands of DNA and not both, so it avoids the formation of double-stranded DNA breaks. The reverse transcriptase domain is responsible for copying in edited DNA sequence at the target location in the genome. So the second key component is the prime editing guide RNA or pegRNA. It's shown in orange. And this pegRNA has two sort of programmable or tailored components that are specific to the mutation that's going to be corrected. The first segment of that is indicated here as the search sequence. So this programs the editor to the location in the genome where the mutation exists. And the second modular element is the replace sequence. So this is a template for the corrective edited sequence that will be incorporated in the genome, and it's copied in by the reverse transcriptase enzyme. So the sort of components here that we are able to swap out allow us to target many different regions in the genome and also make many different types of edits. Because the DNA polymerase is copying in from a template, we can use this mechanism to correct point mutations, small insertions, and small deletions. So it's a very versatile editing platform. And because the edits are templated by nucleic acid, this editing mechanism avoids sort of other bystander type edits that it can occur with technologies like base editing, and the avoidance of double-stranded breaks limits the sort of indels or other byproducts at the target locus in the genome. Additionally, the fact that prime editing avoids double-stranded breaks also likely leads to the minimal or no detectable off-target edits that we and others have seen or observed when evaluating prime editing systems. Again, because of this sort of modular pegRNA component of the prime editing system, we can in principle use prime editing to address many different mutations across a particular disease indication, that different mutations that might be found in different patients. And as many of you are aware, some diseases have very small numbers of mutations, and other diseases have a large diversity of mutations that exist in the patient population. So some of the beauty and the sort of magic of these programmable gene editing systems is we could tailor these agents to particular mutations and personalize the therapies. So next slide, please. So really one of the most exciting prospects, as I mentioned, of these technologies is the potential to make personalized gene editing medicines. And I think many of us in the gene editing field hope to see this vision come true one day, but the reality is we're currently at a place where developing gene editing therapies for ultra-rare mutations is really prohibitive from a cost standpoint and a development standpoint. So as a result, where we are right now is that we're focusing on a small subset of prevalent mutations or relatively common mutations where there are enough patients to treat. However, we do believe that this is sort of on the path towards realizing the ultimate goal of bringing these therapies to more patients. So we believe that we can start with these more prevalent mutations and establish the platform, and then in the future leverage a lot of the data from these initial endeavors. So for example, the CMC and non-clinical studies, and use that to develop in the future therapies for more rare mutations, and thereby hopefully streamline the regulatory process to that ultimate goal. Next slide, please. So I'll be touching on a lot of challenges that I think have already been brought up today. So I don't want to be too redundant in iterating these, but we're broken down into a few different areas. So non-clinical CMC and clinical challenges. In the non-clinical sort of area, we see that the challenges exist in making tailor-made animal models for each specific mutation. As you can imagine, as you scale this from a couple mutations to hundreds of potential mutations, making an individual animal model for each of these would be prohibitive. We also see a lot of challenges with doing redundant biodistribution, reprotox, germline editing, and GLP toxicology studies when we're keeping the delivery vehicle constant, for example, a lipid nanoparticle system. And also the exhaustive off-target risk assessment that's done for each mutation is very resource intensive and frankly also prohibitive. So we see areas to address some of these challenges, and I'll kind of touch on those at the end of our presentation here. We also see many challenges in the CMC area, both manufacturing processes and analytical assays that need to be developed for each new editor. I'll focus on a little bit on how the analytical assays could potentially be streamlined. And finally, there are challenges with clinical dose selection in the context of small patient populations where sort of a dose escalation is not as reasonable. And we think we could leverage prior clinical studies to choose doses for patients that receive therapies later on for their rare mutations. Next slide. So we're using today, as in our example, our liver-targeted lipid nanoparticle system for prime editing. And as many of you are aware, a lipid nanoparticle it can be used to deliver RNA components in vivo, in particular very efficiently to hepatocytes in the liver. And our lipid nanoparticle contains 8 different components, various lipid components, targeting ligands, as well as the prime editor mRNA, and then 2 components that can be varied depending on the mutation that's being targeted. So both the pegRNA and the nicking guide RNA. So across different prime editing therapeutic agents that address different mutations, we imagine holding 6 of these 8 components constant and changing just the 2 guide RNA components. So thereby holding a lot of that delivery vehicle the same, especially what is outside on the surface. And because we see evidence, and as of others, that the lipid nanoparticle vehicle largely drives the biodistribution and the toxicity of these systems, we believe that this is sort of going to be something that we could leverage once we gain enough data and enough experience with these systems. So next slide, please. So in addition to the nanoparticle itself, the RNA components that we use for prime editing are also quite modular. As you can see for the prime editing guide RNA and the nicking guide RNA, the regions shown in blue there are typically held constant across different guides, whereas the variable regions are shown in orange, green, and red. And so a large percentage of this RNA sequence is actually constant even as we change these guide RNAs to address different mutations. The prime editor mRNA in many cases will be held exactly constant. There'll be no changes at all. However, we do imagine potentially in some cases making small changes to that prime editor protein that make editing of a particular mutation more efficient. So for example, changing the PAM specificity of the enzyme. However, this typically requires very few changes to the editor protein and to the mRNA so that the mRNA remains 98% or higher identical to any other prime editor mRNA sequence. Next slide, please. So 2 areas where we have applied our liver-targeted LNP for prime editing are Wilson's disease and glycogen storage disease type 1b. You heard about glycogen storage disease type 1a earlier from Beam. So this is a related disease, but caused by mutations in a different gene. So both of these diseases are caused by loss of function variants in genes that predominantly show their effect in the liver. And while Wilson's disease is a relatively common genetic disease with about 20,000 patients in the US and EU, glycogen storage disease type 1b is much less common and also doesn't have as many prevalent mutations as Wilson's disease. In Wilson's disease, there's a relatively prevalent mutation, the H1069Q mutation. So you can imagine addressing Wilson's disease prevalent mutations would be a bit easier than addressing mutations in glycogen storage disease type 1b. And we'd like to think of ways that we can move from both the common mutations in Wilson's disease to the more rare mutations within Wilson's disease, but also to other indications like GSD1b, where there are few prevalent mutations. So, we envision a path that looks something like this. So again, starting with development and doing all the work and collecting all the data, including clinical safety and optimal dose selection for our LNP delivery system of prime editors for common mutations. So example, again, being that H1069Q mutation in Wilson's disease. And then later on in a sort of subsequent phase or progression, demonstrate that a lot of these different components are modular, collecting data again to support that using these systems in additional mutations generates a similar outcome. And further taking that to now more rare mutations, and then also again to different disease indications where there are not a large number of patients to start with. Next slide. So to touch on some of the challenges here, here's as a first example. So we've made 4 different animal models in these 2 different diseases. So 2 different mutations for Wilson's disease and 2 different mutations for GSD1b indicated at the bottom there. And we've developed prime editors that can address each of these mutations. And you can see from the data, we've been able to develop prime editor LNPs that can correct these mutations between 80 and 90% in vivo in hepatocytes. So the question becomes here is, what do we do now when we want to address the 3rd mutation or the 4th mutation or the 10th mutation? Is it feasible to generate a new animal model every time we want to create a prime editor that addresses a new mutation? And we do think there is a high degree of biological plausibility that correcting mutations, at least in these particular diseases, even if they are different, would restore the gene to the same wild type sequence that would have the same function and have the same, as long as the expression level or the correction level was consistent with other edits. So we believe that this is a potential place to focus on streamlining preclinical efforts by utilizing in vitro cell-based systems, as well as surrogate editors that can be used for correlating in vivo and in vitro results. The next slide. So the shared editing system and delivery vehicle also lends themselves to platform-based preclinical development, and we've heard about this before already, but to sort of quickly go through it, we think that across pharmacology, DMPK, and toxicology, we can leverage a lot of early data and apply it to subsequent prime editor agent development. This includes leveraging biodistribution, germline transmission, GLP toxicology, and other studies that can we believe will be similar across different prime editor LNP products. So once these parameters are established, we really would like to see that be leveraged for subsequent mutation-specific editors. Next slide. So we've developed a comprehensive suite of assays to nominate and also detect potential off-target edits made by prime editing. This includes both local off-targets at locations in the genome that resemble the target site, as well as larger structural changes or chromosomal scale changes, such as translocations or large deletions. And we've felt that this is important to do for the initial products that we would aim to develop for prime editing, to really gain as much data and understanding of the off-target profiles as we possibly can. But in the future, and in particular for ultra-rare mutations, we think that we can take a data-driven and mechanism-based approach to simplify this risk assessment and streamline development of editors for more rare mutations, and really focusing on high-yield assays that that focus in on the most likely off-target risks and avoid some of the more redundant and sort of unsupported assays in terms of what would make for a safer product. Next slide. And here are some data that we have generated actually for our CGD program. This is evaluating the off-targets and structural changes of our prime editor in this program. As you can see on the bottom left, we've evaluated over 500 different potential off-target sites in the genome and have not identified any significant level of off-target editing at those sites. By contrast, if you were to use a Cas9 nuclease, you would identify multiple off-target events as indicated on the right. We've also looked for large deletions and translocations with these prime editors, and we do not observe any. And again, by contrast, if using a Cas9 nuclease, you do observe a substantial amount of the translocations. So we just believe that this supports the notion that you should take a mechanism or editing approach-based risk assessment for off-targets and not treat every sort of a gene editing system the same in terms of how you consider what are off-target risks. Next slide. So from a CMC standpoint, obviously these prime editing LNPs are quite complex drug products. They possess multiple components. And so meeting the regulatory expectations for manufacturing processes and for assays is quite burdensome in the context of rare diseases when each prime editor with a different guide RNA is considered sort of a new drug substance and a new drug product. And we don't think that necessarily all of that additional work that would be done would actually add additional assurance to the quality of these products. So, taking the analytical assays for the drug substance and drug product as an example, currently we are requiring 40 assays or so for the different drug substances and another 25 for the drug product. And we'd propose that a risk-based reduction in the number of these assays be taken, maybe starting with the orthogonal assays to eliminate those and eliminate some of the redundancy. Next slide. And just lastly, to finish up with the sort of clinical modularity, we believe that we can also leverage data in a clinical context to inform dose selection for subsequent prime editing agents. So again, using our example of Wilson's disease and the H1069Q mutation, we could imagine doing sort of the traditional dose finding in the context of that prime editing agent. And then for subsequent mutations within Wilson's disease, and maybe even in the future in other liver-targeted LNP indications, utilize those doses for early first-in-human dosing for those rare mutations where doing a dose escalation study would be less feasible. And next slide, I'll just wrap up with sort of summarizing some of the regulatory challenges that I've touched on today, and also some of our proposed solutions. So I think this really captures everything that we've discussed today. So one of the challenges that we've seen is that diverse gene editing mechanisms exist, and this includes nucleases that generate double-strand breaks, base editing, and prime editing. And we'd propose that a mechanism-specific risk assessment be taken for each of these modalities, and not necessarily lump them all together. So, for example, for prime editing and base editing, one might focus less on double-strand break toxicity and on other aspects and instead of double-strand breaks, given that they don't generate those intermediates. A second challenge is, creating the animal models for each mutation. That is not feasible as we scale to multiple mutations. So we propose that we'd establish potency by correlation to benchmarks, both in cell lines and in vivo, and use cell lines primarily to assess correction of mutations, and then use surrogate editors instead of tailor-made animal models to demonstrate activity in vivo. As a third sort of challenge, we think there's a high burden of non-clinical studies as we change the guides to target different mutations, and we don't believe that the redundancy of these studies would actually add that much in terms of additional data that would provide higher confidence for safety. So within the same delivery platform, in particular, the liver-targeted LNP, we believe that we could leverage data for the biodistribution, reproductive toxicology, germline editing, and GLP toxicology from earlier studies. In terms of assessing the off-target risks and the genotoxicity, we, again, to reiterate, want to propose a mechanism-based risk assessment and simplify assays for later products and maybe focus more on in silico approaches to reduce sort of the burden of doing that evaluation. In terms of the CMC challenges, we think that recognizing this as a sort of platform-based CMC would be advantageous once the editor and the delivery system have really been established and data has been generated. Changing the guides, we believe, is a minor change. So leveraging sort of the process data, the assay qualification, stability data, and really focus on just the few components that are critical in those later products. In terms of the clinical challenges, again, dose escalation and safety studies for small patients populations is challenging. So we propose to expedite or eliminate even the need for dose selection in those particular ultra-rare variants. And then lastly, I didn't touch on this earlier, but multiple regulatory authorities exist, and they each have distinct expectations for what they want to see for regulatory filings, and this increases the cost and complexity for the filers. So we would propose that the agencies harmonize and align on common expectations here, align on terminology and format to reduce sort of the effort required for these submissions. So with that, I'll end and happy to take any questions.
Thank you, Andrew. Any questions?
One question here on the floor.
Yeah. Yeah, thank you for some interesting presentations. Very, very nice. This is actually a question to all three of the presenters, I think. I mean, I understand it's quite a challenge on the regulatory side here. And I don't know, I think some of you have experience both with the FDA and the EMA. I don't know if you've all been to scientific advice procedures, for example, at the EMA, but have you had parallel discussions with the FDA and the EMA on some of these issues? For example, requirements for non-clinical studies or how to design clinical studies, etc. I mean, we do have the possibility to have parallel consultations with the FDA, and I have the feeling it's an underutilized possibility that developers don't use that much. But I think it would be useful, maybe for you, and also it's useful for us, I think, as a regulators because it gives us a opportunity to sort of see common grounds with our FDA colleagues also. It's more of a general question to you.
Yeah, I can't speak to that myself, but I can see the benefit that you're proposing here.
If the other speakers are still on, any experience there you want to share with parallel scientific advice?
I can just comment quickly. To my knowledge, we haven't actually had parallel discussions with FDA and EMA, but I think, like Andrew commented, I could see the value in some of those conversations. We've certainly had separate discussions with both FDA and EMA, where we have, received different advice and guidance. So maybe there is an opportunity through some of those mechanisms to help with the harmonization.
I think Pam has a question.
Hi, sorry. Yeah, I will talk about that in my next presentation because we have done parallel filings, so maybe we can dive into that.
All right, thank you. Good segue. I think we do have one more question though, maybe two.
Yeah, so it's again from the room. I had a question with regard to if you do, for instance, a bigger filing with more patients, so with a mutation that's more prevalent at first, and then come in with kind of, just thinking out loud, with smaller amendments to the dossier, for instance, for the other mutations, what would be the minimal number of patients that would be or would result into sufficient clinical support for an application that is kind of the umbrella that you would use to add other small mutations to?
I think that would depend on the context of how maybe rare those mutations are and how many patients, feasibly can be identified with those mutations. Likely that wouldn't be, the first step after maybe choosing the most common mutation is probably going to be choosing the second most or third most common mutation. So going in order of prevalence. And I don't imagine it would be a rapid drop off in the number of patients that we could identify. But I think it really just depends on that particular disease area and the sort of prevalence of the mutations in that population.
And with a follow-up question, would you use, for instance, then for the coming mutations, a personalized cell system for proving efficacy, for instance, like organoids? Would that be possible for some diseases, or what could you think of that we could see as an efficacy system in vitro?
I think there are a number of options, and I think it depends on maybe on the target tissue that you're going to be doing the editing in. Organoids could be one possibility, but I think simple cell culture, cell lines is another option. And what I would think is the right path forward is to use a data-driven and scientific approach to correlate the editing efficiencies that you observe in these cell lines to what you would observe in vivo with known standards or benchmarks. And once you've established systems that reliably can sort of predict the efficiency or potency of the editors, move forward with those. So I don't think I would be prescriptive necessarily at first, given that we would be focusing on a common delivery modality like a lipid nanoparticle, and where we don't think that would vary substantially across different editing agents that target different mutations.
And another question, so would be the correction level be sufficient, or would you think that functionality of the gene would also need to be included as an efficacy marker in vitro?
Yeah, I would hope that the earlier examples, so for the more common mutations where we've generated animal models and demonstrated correction of those mutations and subsequent sort of phenotypic correction, I would hope that given the diseases we're focused on are loss of function diseases that have sort of well-established biology. I heard the comment earlier, and I appreciate the comment that different variants might have different effects and different severity. But for these particular diseases that we've discussed, we think that really it's just establishing a certain level of functional gene expression is what's required. And so if we can surpass that threshold, which could be established again with data and using sort of other support, I think that would, in my opinion, be sufficient. And so you could benchmark based on editing efficiency, because fundamentally the beauty of gene editing and correcting mutations in situ is that regardless of the starting mutation, you will be left with the same product, which is a corrected healthy gene. So viewing those again with I think high biological plausibility, viewing those as the same end product, we'd hope that you would recognize that as achieving a similar level of sort of phenotypic correction if you can establish that you reached the same level of editing.
Thank you.
Thanks, Ed. We have time for one more very quick question.
Yeah, thank you. Very quick comment. I keep hearing about the platform issue from our presenters. I don't want people to go away with the idea that until we have a definition of a platform or requirements for a platform that things like prior knowledge cannot be used, because we've been used to accept that for a long time for other category of products, certainly for the CMC and probably for the non-clinical as well, but I'm more from the CMC side. So please keep in mind that you don't need to repeat many of the testing or development that you do for one product if the product doesn't change in one of the components. So I just want to clarify that and let you know that if you come for scientific advice, you'll probably go away with a very good outcome from us. Thank you.
Thank you for your comment.
Thank you for that clarification. So with that, we will move on to our final speaker in the open session, Pam Stekiewicz from Arbor Biotechnologies. We'll talk about challenges in gene editing drug development.
Thank you for inviting us. I'm going to present on the behalf of Arbor Biotechnologies. As Mike mentioned, my name is Pam Stekiewicz. I'm the Chief Operating Officer here, and I'm accountable for the development line functions, CMC, quality, regulatory tox, and project leadership. I've been in the industry and trying to develop gene editing medicines since 2016. Started at Editas, went to Flagship, and have been at Arbor for over 4 years now. I wanted to give you a presentation from the industry side about the challenges in developing these drugs. I think we all believe in the promise of the drug and how important they are, especially to patients with rare and ultra-rare diseases. So we'll just walk through a couple of the drug development challenges we face, walking through preclinical CMC and the need for early global clinical trials. We'll prepare and share a summary of our findings to date. We're using real-world data, and then recommend some processes that may be improved for EMA to take into consideration as they develop future guidances. So a little bit about Arbor Biotechnologies. We're a second-generation genome editing company. We were founded in 2016 from Feng Zhang and David Walt. We were founded on the principles of AI machine learning. Our goal was to mine the metagenome to find nucleases that were outside the Cas9 family. To date, we have 10 proprietary nucleases. These nucleases have different PAM binding sites that allow us to access over 90% of the human genome, where Cas9 is restricted to about 10% of the human genome. These nucleases have staggered end cuts, so they behave slightly differently. Since we are a company with the focus of driving programs into the clinic and ultimately to the market, we really wanted to focus our early portfolio on programs where we could target the organs. So we're concentrating on targeting the liver and CNS. Our lead program is ABO-101, which is for primary hyperoxaluria. This is a rare disease in which patients overproduce oxalate, leading to massive kidney stones and kidney failure. This recently entered the clinic earlier this year, so I'm proud to say we're a clinical stage company. We also have our nucleases. We've had great advancements in being able to miniaturize these nucleases. So we can fit the nuclease with 1 or 2 guides and a full-length promoter into an AAV. We feel this is differentiating for us, and we want to tackle devastating diseases. We're starting in ALS. So just big picture, just wanted to point out some, I think everybody knows here, but some of the big picture phases of drug development going from research, IND enabling clinical trials into market authorization. Some of the differences we see in gene editing on the early stage of the pipeline. We can move relatively quickly through discovery and lead optimization phase. This is because there's only so many options we can to tile the genome and find unique guides. This is very exciting and can be great that we limit this, but then we have to prepare for development stages sooner. And so if we talk about IND enabling, some of the key differences we've seen and talked about today is tox. It's a possibility we can go into one species. Usually it's NHP. We concentrate on genotox, specifically off target, been discussed a lot today. We also want to understand biodistribution and potential on-target editings in unintended tissues. And what I find really interesting is CMC is so colossally outweighed in the cost of doing CMC for gene editing programs that there's not much tolerance to kind of consistently iterate on the CMC product. So early in development, we get pretty close to the final product configuration. And of course, in clinical studies, the big difference is that we want to go directly into patients if we're editing somebody's DNA. We go right into patients and we follow these patients for 15 years. So this is totally an industry perspective. As we think about building out a portfolio of programs, we obviously look at the patient population and unmet medical need. We want to understand if there's tractable biology. We will lay out drug development plans and costs. And at the end of the day, we have to say there's commercial justification and that we can get pricing that would commensurate the expense in development. So I think if we look at this reasonably, I think everybody can realize that it's really the small biotechs that are going to be in the rare and ultra-rare disease space. Big pharma is not going to go after patient populations that are in the low thousands. Even if they can get a $2 million price tag on therapy, they usually set a bar with an NPV analysis of $1 billion or $1.5 billion, and they're not going to make that kind of money. So it really depends on biotechs to be operating in this space efficiently. So if we want to operate as a company, we have to go to investors and lay out all these plans in our portfolio and justify that we can gain investment and we can bring programs to clinical proof of concept. I think we've all seen that the investors are not rewarding platform type companies, that they want to see clinical reality and data that yields more investment. So we need to have investor funding and we need clinical data to either raise more money, partner these assets, or get acquired. So I think the biggest challenge we have is time and money. And if you look at public data or data on public companies in the gene editing space, there's about 8 or 9 of them, you'll see that they burn anywhere from $25 to $100 million per quarter. So this is a colossal expense that you need to raise money in order to get to that clinical data. So we've talked a lot about preclinical, so I will just highlight some of the areas here. Our goal is in preclinical to demonstrate that we have a favorable benefit risk profile for patients. We have a variety of models that span from in vitro, in vivo models, patient-derived cells, and other things that we can correlate editing amounts to modifying disease phenotype or disease outcomes. At the end of the day, we can use all these models. But as I mentioned, we want to go into that first clinical trial at a minimally efficacious dose. So one of the challenges we have is taking this modeling data and really trying to estimate what that benefit will be to patients in that very first patient, that very first dose we give, and have a reasonable dose escalation, and of course, always maintaining safety. On the risk profile, we've talked a lot about assays that have been developed to date, and they're getting more and more exquisitely sensitive to measure things like off-target on the human genome using cell, organoids, or other assays. In general, what we've seen and what we've noticed is we have a lot of tools to measure all these things, but there's a bit of a gap about how you interpret the biological risk, whether it's an off-target hit. Sometimes we'll get these assays, we'll get the results, we have super low levels of detection, and at the same time, it's a paper exercise to infer what that risk is. Sometimes the off-target gene is of unknown function, or it's in a non-coding region. So how do we really assess the risk? And we've talked a lot about biodistribution. It's mainly dependent on the delivery modality. And if you have on-targeted editing outside the tissue of interest, how do you interpret this risk? And GLP tox, everything is a model and a tool, but sometimes we can't understand things such as immunogenicity from some of our tox models. So we move into CMC and some of the challenges we face here. You've seen this all in different iterations, but here's just a illustrative process for an LNP. And some of the challenge we see is we have the raw material, starting materials. We have different drug substances that are encapsulated in a lipid nanoparticle. We then go through purification and filtration, bring it to the desired dose, and vial it as a drug product. I think people understand that this is expensive, but I'm just putting some numbers down for an understanding that that first batch of LNP may cost anywhere from $7 to $11 million, and it takes about 20 months to prepare for the whole process end to end. The EU has always had strong concerns or feedback around CMC, and they have some unique requirements around raw starting materials, impurities. They have requirements around release specifications for the drug substance, the drug product that may be unique to other countries, and requirements around potency assay. This all leads to additional method development, assay development, and validation, in addition to additional documentation. And just for some context, as we're building up to slot in manufacturing slots, we got to back in validated release specifications. And this is a very time-consuming and costly effort to get these assays validated. We found that scientific advice, we went to meetings, many of these meetings in preparation for CTA IND filings, and these are a great place to align on specifications and make sure everybody is comfortable with the path forward. But in order for us to get binding response and minuted advice back from agencies, this meeting comes pretty late in the drug development process. So I think just for context, if they come back and say you need additional assays or things like that, it could lead to substantial time delays in manufacturing. And really worst case scenario is we miss a manufacturing slot because we're not ready to manufacture if we need additional assays. So a bit about some of the things we found that were a bit unique to EU CMC. Prior to IMPD submission is around GMP master cell banks being required where they're not necessarily required in the US for phase 1-2. The cost of getting a GMP plasmid master cell bank is greater than $1.5 million. As I mentioned, the unique requirements for some of the raw starting materials and impurity requirements and release specifications that are a bit more stringent than rest of the world. Just for context, if we are going to produce at-scale batches, engineering and GMP batch for filing, sometimes due to the EU regulatory timeline, and then followed up by need to release materials through the QP, and then, which I know is all in your control, things that are outside your control are EU clinical site contracting, can lead to a timeline of not dosing a patient for 6 to 9 months post regulatory filing. And then lastly, just like to talk about expiry or shelf life printed on the label. A lot of times for LNPs, this is a frozen drug substance that's shipped around the world for global trials. Just for context, we're doing real-time stability. We have leading indicator using our engineering batch ahead of GMP. However, every time we need to reprint the label, we need to pull back the drug supply, and it costs over $100,000 per label rerun. So some of the findings we had post-IMPD filing, when we went to the EU, we filed so we did file in parallel. We filed in EU, UK, and FDA within a month of each other. Interestingly, IMPD feedback for, we had 60 additional requests for additional information, while the UK had 9 additional requests, and the FDA on Module 3 only had 2 additional requests around CMC. So that's just for some context about just the different perspective on CMC. QPs in the EU and the UK also have asked for additional sets of requests that happen post authorization. So this was challenging. We had over 60 questions from the QPs and post being authorized, which led to delays of about 3 months. So all in all, CMC, I think, is viewed differently in Europe, and maybe we could talk about how to harmonize this with the rest of the world. A little bit around global trials. In order to reach rare and ultra rare disease patients, we really need to file in multiple sites across the world. Even if we're a small stage company, we need to do this. This is pretty intense for a small company. We found that there were definitely strengths and challenges in the EU when we did this. The scientific advice we went through PI was very valuable to us and gave us great feedback. We also appreciate the predictable timelines for assessments and clock stops. That's very transparent, so that we could plan and set expectations and allocate our resources. The clinical trial information system, the single submission, was very convenient, and it reduced the duplicative activities across multiple countries. We appreciate that, and there was also transparency there. Some of the challenges we faced is that we had unharmonized feedback on part 2 versus part 1. And I think Christy mentioned it in Intellia. The problem with if we do any changes, we got to go back to the ICF and translate in multiple languages again. And so this unsynchronized timing led to 3-month delay in order just to address simple questions. There are tight timelines on RFIs, and as a company, we accept that and we take that accountability. However, when you ask us to get input from vendors or other people, it's difficult to align with the timelines that may be very short. And then in addition, this translation piece of it is relying on outside people to then translate all the corrections into the documents. So here I just give some real world examples of timelines from submission to clearance approval across US, UK, and Europe. And you can see that Europe has extended timelines compared to other regions. And it's back to the basic principle that our biggest challenges are time and money. So we burn money every day that we wait to enroll those clinical patients. So in summary, we really value EMA input. It's really important to us. You have great scientists and understanding of the complex manufacturing for these complex therapeutics. And really importantly, we want to ensure patient safety, and we value the EMA input and experience around helping us devise the best clinical plans. I will say that due to the GMP requirements compared to rest of world, as a small company, we really need to carefully consider if EU is essential for clinical enrollment in early clinical trials. If it is vital, then we really, because due to the timelines that we need to validate assays and plan so far in advance, we really have to have rest of world meet the EU release specifications. And this just costs us more time and money up front. I think part of the problem is there's such a fear that you're going to get up to a scientific advice meeting and find out you need to do something additive, that we front load a lot of assays that may not be essential in order to understand the product. We believe harmonized country filing is a good faith effort, but in actuality, there's unharmonized feedback that can cause time delays to open clinical sites. And the EU regulatory timelines are long, and the company burns money. So if we could recommend a couple things for your consideration as you think about new guidance. On the preclinical side, other people have talked about it, but one of the things with type 5 nucleases is, since we have a bigger staggered deletion of 5 to 20 nucleotides, we have an exquisitely low level of detection of off-target. And so if it's detected, it's flagged as a risk. And I think that's probably something that could be considered in understanding what that threshold should be. We would also appreciate any guidance on off-tissue editing risk assessment. And when we think about clinical, if we could enable submissions to update the ICFs to align with requested protocol changes, that would really limit any time delays. On CMC quality, I think other people have also expressed similar requests to revise requirements around raw and starting materials that are not necessarily in the final product, to look at CQAs to understand what's really needed for these early batches. And I would, express that you, as a regulatory agency, see much more materials and could really understand this better than any individual company. Consider allowing for non-GMP plasmids and master cell banks for early clinical studies until we understand if there's any efficacy. If we, as I mentioned about the GMP, if we have file on GMP material and we don't use it for 9 months, but it has limited stability, we need to plan for another manufacturing run almost immediately after we start clinical studies in order to have continuous clinical supply. So we would ask that you consider filing on engineering run for stability limited ATMPs. Maybe something a bit controversial, but we'd like to consider streamlining or omitting the QP requirements post CTA authorization and limit the number of PPQ batches for ultra rare and rare diseases. On the regulatory process side, I think to align the CTA IMPD review timing that's consistent with US and UK, especially when we have to do these global trials. It's hard when we get approved in one place 1 month and then another place takes 4 or 5 months. And promote consistent classification of amendments, substantial and non-substantial across member states, and allow for concurrent review of amendments to limit time delays. So thank you very much for hearing our thoughts around from an industry's perspective.
Thank you, Pam. Any questions for Pam or any comments?
I think the issue is there's a lot that can be commented on, but I think there are a couple of fundamental statements that clinical trial approval is national remit. And I think when there is a talk about global convergence, the thing that you should never expect is processes to be shorter, because there needs to be predictability of workload. So the more stakeholders you need to include in a decision, the more times you need in the process. And when it's a single agency, whether it's the UK or the US, it's always much easier to come to a quick solution because you don't have the need to interact on what your conclusions are. So I think that is a fundamental issue. And being a clinical trial assessor, having been a clinical trial assessor myself, I see the importance of having predictable timelines for everyone. The other thing you were referring to, the knowledge we have on different products. Again, the clinical trial stage is in a database that is hosted by EMA, but it's national member states dealing with them. And I think that the one message that I want to convey during the meeting today is we need to share information, maybe product non-specific information of what is the state of the art, what is known, what can we build upon? Because in the end, that is what's going to help everyone. And if everybody reinvents the wheel. But and my last point is, sometimes I think your expectations of what the regulators are incapable of accepting might not be actually correct. So I would definitely encourage upfront discussions, whether it's on the clinical trial level or on the level of EMA. Thank you.
Thank you. Those are all fair questions. And I, as I mentioned, I think it's probably more a fear factor of being rejected late in the end game, especially around CMC, that leads us to front load things that may not be necessary. So I completely agree.
Thank you, Pam. Any other questions? Should we move then to closing remarks?
I think we can do. are quite late or expanded our time already by half hour. So let me give quickly the floor to Ilona Katcher for the closing remarks.
So I'll try to keep it short, but touch on all the points. First of all, a big thank you to ARM for engaging in this activity. A huge thank you also to the speakers, particularly the ones for overseas, for accommodating the European timelines. Thank you to all the participants online, whether in Europe or overseas. I think the clear takeaway from everybody needs to be is that we need to work together to develop meaningful guidance. We need to have a better understanding of the state of the art. The knowledge on gene editing is not evenly distributed geographically. We need to make sure that we share that knowledge to enable meaningful regulation for the sake of patients. And I always want to emphasize meaningful regulation. We definitely need more time for discussion and interaction. And I can assure you that as long as the CAT exists, we will foster and work on improving the regulatory framework for ATMPs. And before the question comes up, no, I do not know what the future structure will be, but I do know that the needs of the network and of the stakeholders are being recognized, and there's a lot of thought of going into what future structures may be. At the moment, we don't know. And the main point is, don't expect regulators to come up with guidance. We need input first. We need to have the experience that developers have gained. We need to learn about the challenges they faced, but at the same time, don't underestimate the flexibility that regulators have, particularly on scientific issues. When it is a legal issue, it's very difficult for us to move. And when it comes to convergence, that further complicates issues because convergence takes time. We have to overcome different legal requirements. But what we can work on is harmonize on the science as a first step. And I think also we need to, in the discussions, make clear whether we are talking about the ability to treat single patients, potentially under legal exemptions, whether we are talking about requirements for clinical trials, or whether we are talking for requirements at marketing authorization. And I hope I've covered most of the issues, and I thank everybody very much. Mike, also thank you to you. And a big thank you to EMA colleagues for hosting today and all the organization that's behind this event.
Yeah, and thank you for organizing. And please just let us know how we can help with any next steps, including promoting information sharing to inform any guidance.
Okay, and then last word from me, thanking all the speakers. First, the participants from our side here in the room who traveled into Amsterdam to follow the discussion in a live fashion, and then all the participants online. I hope this was also of interest for you. And like said, this is start for work for us. And it certainly has been a very informative day today. I thank you and wish you all a very nice evening, afternoon, and hope to see you in future interactions with the committee.